Antibiotics were one of humankind's greatest successes, in my personal belief. We are at war with microorganisms, and microorganisms definitely long-term are winning. Admittedly, most of the humankind's drug discovery efforts resemble throwing screwdrivers at a TV randomly and then finding what tunes it, and most of them don't. It's helpful when the TV is not the only TV you have. The moral of the story is it's a complicated world. We are complicated things that live in a complicated world. The biggest mistake that we as people can make is oversimplifying. Treating things and people as objects, especially people, is one of the worst mistakes that one can come up with.
In this episode of the 632 Nanometer podcast, the team takes a deep dive into industrial drug discovery and biophysics with Artem Evdokimov. A true renaissance man in pharmaceutical development, Artem earned his PhD from the Weizmann Institute of Science and has been developing breakthrough drugs ever since. His journey spans from the National Cancer Institute to industry giants like Pfizer and Monsanto, and more recently to leadership roles at cutting-edge biotech startups. The conversation ranges from the fascinating history of insulin to the complex biophysics of Ozempic and other GLP-1 peptides, with Artem breaking down some of the toughest challenges in modern drug discovery. Fair warning, you might want to brew a strong coffee for this one. The discussion gets deep into the scientific details, offering a rare glimpse into how modern medicines are actually developed.
For the person who is first time listening about drug discovery, what should we start with? How do we get to this complex topic?
There's no good answer to that question, so I'll just give you a bad one. You start by being fascinated with the way biological systems operate. And biological systems to this day remain some of the more complex entities that you're going to experience in your daily life. So your own body is probably the most complex machine that you routinely touch. It has more moving parts, it has more systems in balance than anything else that normal people would experience, rivaling the hardest and the most complicated supercomputers that we ever built. At some point, these will exceed in complexity at least one cell, but right now they're all below one cell. So the way to start in drug discovery is to become fascinated by how biological systems operate and also be ready to mess with them. And admittedly, most of the humankind's drug discovery efforts resemble throwing screwdrivers at a TV randomly and then finding what tunes it, and most of them don't. It's helpful when the TV is not the only TV you have. This necessitated the transition from experimenting on oneself to experimenting on other people, whether they like it or not, and then to experimenting on animals, and then later on cellular systems and isolated biological molecules. But the important bit is you have to be committed to an understanding of something, and also in your desire to change it, you have to be committed to many failures. Because the story of drug discovery overall is a story of fantastic successes, very, very strongly interspersed with massive failures over and over and over again.
And how does the story start? Is it 100 years ago, 200 years, or even longer?
Thousands. So one could argue, and we can have a nice argument, what the first drug ever was. So I suspect that the first drug ever may have been either alcohol, cannabis, or opium. It's hard to say. But animals are known to ingest certain things when they don't feel good. A classic example is dogs eating grass to vomit when they eat something not so nice or when they're burdened with parasites. And in a sense, you can consider birds sunbathing in dust to get rid of their parasites to be a form of drug. So first drugs very likely were, like I said, let's just start with alcohol. So people realized that those who drink small beer or diluted wine, they don't suffer as much from various diseases, intestinal and otherwise. And so the tradition of brewing alcohol has taken off, not just because drinking alcohol is fun, and you get intoxicated, but also because you actually get healthier in the long run, provided that you're not an actual alcoholic. I mean, if we're talking just preventative function of alcohol as a antiseptic. That starts probably thousands of years ago, and definitely already ancient Egyptians had pills, although those pills were very different from what we consider pills. And in fact, the word pill comes from us from Greco-Roman period with the foundation of the word pilula, or pilula, as some of us might recognize. But those pills were pretty neat. They were made out of clay, and they had goat blood in them as a binding agent, and they had all sorts of things. Famous story for an ancient Egyptian birth control medicine containing, among other things, crocodile dung. And it was a suppository. You could see where people experimented a lot. What now is considered to be recreational drugs for a lot of people were life-saving medicines at the time, including things like cocaine. So chewing coca leaves for anesthesia was likely to be very important because already folks who lived in the region were practicing trepanation, surgery of various kinds, even cataract extraction probably. And those are not fun procedures to begin with. And then if you're doing them without anesthesia, it's really super not fun. There's a number of pointers to the development of proper drug discovery, and they all kind of start in the 1600s, where people were experimenting with ways to put medicine into a human body. We owe a lot of drug discovery progress to the invention of what now is considered to be a staple of medicine, which is the hypodermic needle. Right, so how did such a thing become reality? In the beginning, people were experimenting with quills. So you take a goose feather, you chop off the head, you chop off the back, you make a nice sharp needle, and then you attach, for example, a bladder, like literal bladder, from the same goose to the end of it. You fill the bladder with substances, and then you stick it to some unfortunate, hopefully an animal, but probably a person, and you squeeze the bladder and see what happens. So for the longest time, people were experimenting with this kind of stuff, trying to understand, even on the most fundamental way, is eating a medicine the same as injecting it? What are the differences? At the time, they didn't know. Again, remember, 1600s, 1700s. And culminating in 1844 by an Irish doctor, Francis Rind, who actually perfected the process of manufacturing appropriately shaped hypodermic needles by rolling a piece of steel and sealing it on the edge. The first syringe-looking devices were actually invented a lot earlier than that, but they were used not to inject things, but to extract things. So in ancient Greece, if you had an abscess on your body, you could have a person pierce it with a syringe-looking device and suck the pus out. Lovely. It's great. Ancient times was so good, so fun. And so you could have that. And then in Victorian England, there were lovely little syringe-looking devices that people used to stick in their unmentionables, and they were filled with patent preparations that were designed to combat things like gonorrhea and syphilis, to a questionable extent. But even to this day, if you excavate near the Thames and you go through layers of muck accumulated in there, you will find these tin syringe-looking things in giant numbers, because that's what people were doing, because venereal disease was rampant and people didn't like it, of course. 1844, however, is when things get interesting. Since we're talking about drug discovery as a process, we have to mention Claude Barnard, who is a French physician and physiologist. It's a very interesting character. He was a crazy workaholic. He held several positions at the same time. He was like a member of the National Academy of Sciences of France, and he was a member of the National Gallery. And he had a number of positions simultaneously. He filled his journals with endless notes. And one of the things that he invented or discovered is the concept of homeostasis. So he was one of the first people to understand that a human or an animal is in a macrocosm of external environment, but you also have a microcosm that is in a state of homeostasis, which is dynamic equilibrium. He also discovered, among other things, and this will get us closer to some of the examples that we can talk about today, he discovered that if you take glucose and you eat it versus if you take glucose and you inject it, your effects are very different. If you inject glucose, you can actually send someone into a hyperglycemic shock and kill them, but you can eat the same amount of glucose or much more. You can eat, I wouldn't say a pound of glucose, but a lot of glucose, and that will not happen. So this was a foundation for what is going on. And so again, we're talking 1845, and now it's 2024. What does this road send us on? So if you remember later, we can talk. But the first medicines were indubitably plant, mineral, and animal origin. This is where it started. For example, spider venom, bee venom, snake venom, they were all considered to be medicines of various provenance. Now, not all of them were actually medicinally useful, but they were interesting in their own way. Things like mercury and arsenic, traditional Chinese medicine, the immortality experiments that they were doing.
I know that like today people get bees and use them to inject their like rheumatoid arthritis.
Yep.
And that seems to work for people. So there's something in bee venom it's a biologic. It's like a protein in bee venom or something.
So
like
bee
that.
venom contains a peptide or a small protein called melittin, which is, among other things, what gives you that lovely sting and the inflammation around the sting. And when you have an inflammation elsewhere, if you essentially overwhelm or distract your system, and deplete these messaging cells that are sending these inflammatory messages, you get relief. It's essentially a principle by which hot pepper plasters work or mustard plasters work. You apply them, they deplete your ability to feel pain to a degree. And so it's a pain of its own, but it's distributed and diffuse. And so it sort of helps alleviate the sharp pain that you might feel. They also cause blood to permeate the tissue because inflammatory process brings blood, and that tends to help with healing. So it's certainly not nonsense. It does help to a degree. There are, of course, also chemical medicines that one can use. These are, not always available. Same with leeches. Hirudin is a known anticoagulant, and people use it.
Leeches produce an anticoagulant?
Mm-hmm. So when they bite you, they produce a number of substances. One of them is a painkiller, so you don't feel the bite that much. And then they produce an anticoagulant, so the blood keeps flowing and they can eat it.
Is that different than heparin?
Yes, heparin is a derivative of a crab shell, and it's basically a sulfonylated polysaccharide. So it's different, yeah.
Oh, okay.
But both are anticoagulants, and both can be used for various...
Is there a reason heparin is an anticoagulant, or it's an accident?
This goes to the core of the problem. When you discover heparin or when you discover morphine, it's always the same question. What is it? Why is the crab made of anticoagulant? I think in this case it is definitely an accident. But with morphine and other things like papaverine and so on, that's a little bit harder because the plant makes so much of it. It especially makes it around the seed pod, which appears to be a way to protect it. So it seems like it's a protective measure against insects and other predators. So things that like to eat it get stoned and go away, and hopefully they spare most of the plant as a result. Or they get poisoned, toxified, and then they remember that eating these doesn't do good. So it depends. But as a result, of course, the opium poppy is a highly cultivated plant because of things that you can do with morphine. And again, not the illicit abuse of substances, but just the fact that it's a painkiller. But again, this goes down to, is discovery of morphine the early form of drug discovery? And I would be advocating that yes, it was. People were trying various things for when they felt not so good, and then they discovered things that made them feel better, like aspirin, for example. It's a derivative of a willow bark extract. Willow bark extract is known to a number of peoples independently, Eastern Europeans and Native Americans. And then in the late 1800s, early 1900s, people started refining it, discovered salicylic acid, and acetylated it to make acetylsalicylic acid. And Bayer now is forever remembered as a aspirin manufacturer.
Did Bayer start as a aspirin company?
They started as a patent medicine company, actually, but I'm not sure, so I don't want to lie to you. We can look it up later. But they also manufactured what they used to call pervitin, which is meth, so to make soldiers march longer and be more aggressive and sleep less and so on. Yeah, good old times. So the important bit to remember is the early attempts at drug discovery were kind of random and they were kind of untargeted, but they were effective because there was nothing before them, so it's easy to find the first few. And that was the classic phenotypic screen type drug discovery. That's the beginning of it.
What does phenotypic screening mean?
So it literally, it means many things, and many other people discover different meanings of it. But in this paper, so for example, this is a relatively new, 2016, I think. This is a discovery of first-in-class drugs. So they describe phenotypic screening as something that has no explicit target, but is a systems-based approach to finding medicinal substances by observing the phenotype, the appearance and behavior of living systems. So in the basest form, you can take an animal that suffers from a particular disease or pain, and then you treat it with a number of substances until the suffering stops in a good way, and that's the phenotypic screen. In a more complicated way, you can take a cell line, for example, a cancer cell line that's uncontrollably dividing, expose it to a staggering variety of substances. You can do millions these days in a very high throughput format, and essentially find those where a cancer cell line will not divide any further, but a corresponding, so parallel human-derived healthy cell line will be unaffected. And there is at least one antibody that has been discovered that way. It's in general not true for antibodies. So antibodies tend to be raised against a particular target. So if you have a receptor on cell surface that you know is associated with, let's say, cancer, then you can isolate this receptor, you can express it, you can purify it, you can raise an antibody in a llama or a goat, and then you can develop this antibody to be a therapeutic. But there is at least one that I know of, and it's actually also mentioned in this paper, where it was discovered by raising antibodies against cell lines. So they didn't know. It turned out to be CD45, I believe.
So
Yeah,
when you say
CD52.
raising antibodies against cell lines, you mean they took a cancer cell line, injected it into a goat, just took the supernatant or the cells themselves and injected them into a goat and then got the antibodies out? Or?
That's one way to do it. The other way, and I have to look up, this is the antibody alemtuzumab. So it's CD52. There's two options. You can do that. These are peripheral blood mononuclear cells. So you take a patient with leukemia, you isolate PBMCs, which are going to be very rich in lymphocytes that went awry, right? The lymphocytes that are not doing so good.
So leukemia is white blood cell cancer, lymphocytes white blood cell. So it's basically isolating white blood cells that have cancer.
As a mixture. So they will be both normal white blood cells and sick white blood cells. And then you can inject them, yes. Will it cause generalized immune response in an animal? Yes. And then you have to work very hard to select the antibodies that are interesting from all the other antibodies that are raised. So that's one option.
I think probably some of the people listening to this don't know enough about what an antibody is. Just quickly to fill that in,
Right.
essentially part of the immune system in mammals is producing these proteins that are selected for attaching to some target. And so there's like a variable region of the protein that your body tunes and finds something that will attach to the target, but not to things in your own body. And a very classic thing to do in biochemistry in general is just to inject animals with something and then collect their blood and filter it for things that attach to that something. So you put that something on like glass beads, for example, and you filter the blood through it, and you collect whatever proteins stick to those glass beads, and then you have the antibody.
Right.
It's things like of that flavor. There's many versions of it, but these days a lot of drugs are in that form. They're these like proteins that attach to something and block it or
So keeping in
change
mind
it.
that the early vaccines, and to this day, some of the snake venom vaccines, for example, or antidotes, that's how they're made. But you have to remember that ideally you want to manufacture this antibody, which requires knowledge of how it is made in your body. So an antibody is a protein. It's a product of a chain of events, which starts in the genome with a gene that encodes it, goes to messenger RNA, goes to a ribosome, produces a protein. If you just collect animal blood full of protein, you can use them. It has been done and is being done, but it's not very safe, nor is it very effective. So when you have, for example, a horse that you immunized with, let's say, snake, a crate venom, right? You collect its serum, you filter it, you enrich it for antibodies in various ways. And then if somebody gets bitten by a crate, which is pretty much a death sentence, you can inject that person with the resulting straw-colored little solution, 10 or 20 mils of it. They will be sick afterwards, not just from the snake venom. They will be also sick from the antidote. So it's a question of balance. Do you want them to die, or do you want them to be a bit sick, but then still?
So they're
hopefully
sick
make
because
it?
of the impurities, like the different things from the horse
A
serum.
whole lot of stuff in there, and that stuff is screaming at your body that I'm horse, and your body says, I don't want horse in my body. So all kinds of alarm bells going off, and that gets activated. A much more gentle and an appropriate way to create these medicines is to discover what encodes the right antibody, then produce it in a recombinant organism, and then inject it into people. This actually goes back to 1978 or so, when a fledgling company, Genentech, was working on insulin, and that was the first time that human insulin was produced in sufficient quantities using an E. coli manufacturing process.
E. coli, the bacteria.
Bacterium, right. So a recombinant organism, recombinant in this case means that it received a piece of DNA under the right control elements, and it can express human insulin. Because before then, people were injecting themselves with pig insulin, horse insulin, cow insulin, and that's okay, but not great, because they were all contaminated with things that were potentially dangerous. And especially for people with hyperactive or hypoactive immune response, this could be a problem. However, again, our conversation is meandering a little bit, which is
Yeah. So you were explaining that phenotypic screening is looking at the outcome, not the
the
mechanism.
overall outcome. And I also wanted to mention that if you're talking about selecting antibodies against cell lines, you certainly have other options. So one option is to try and immunize an animal, which may or may not succeed, because you're bringing in so much other stuff, right? So you certainly will be bringing in these factors that you want to address, like CD52, but you also will be bringing basically everything else.
When you say
DNA.
factor, what you mean is like a protein or something that's expressed in cancer cells.
Yep. So you begin by saying, I don't know what's in the cell that makes it special, but something is making it special. I would really like to find out, but also at the same time, I'd like to address it at the same time. And so this is a particular example where the finding and the addressing are done the same. What I don't know, because I haven't read about this antibody, is whether they did it by direct immunization, which I suspect is maybe not the case, or they did it by a process known as library panning. And it's a useful transition to some of the things that we do here at our company. But library panning entails the following. You find a way, and there's many ways to do so, to express, let's say, an antibody in such a way that the DNA expressing the antibody, or perhaps the RNA, is actually attached in some way to the antibody itself. And I'm being deliberately vague because there's so many ways to do it, but one is, for example, phage display, where it's all displayed on the surface of a phage, and the RNA or DNA is inside the phage.
So phage is a
virus.
You have to define
It's
phage.
a bacterial virus.
Okay. I was actually trying to, maybe you're from the same field or studied biology over a long time, and I'm just I skipped it during my high school. If you can bring to some basics of the foundations of in general, like how the drug works, but maybe that would follow after you.
Right. So this is specific to a great degree to discovery of biologics. So most biologics are either proteins or nucleic acids, DNA, RNA, peptides, proteins.
Wait, Artem, let's back up. Okay, so we were at the 19th century drug discovery, and you were talking about phenotypic versus what's the other option? So there's phenotypic versus target-based.
Right. So in the beginning of drug discovery, you could not do target-based drug discovery. You just couldn't because you don't know what the target is. For example, when you take belladonna extract or you take something for your heartache, like nitroglycerin, you just know that my heart hurts. I'm going to take nitroglycerin. I will feel better. I will turn beet red and I'll have a headache and some other things, but I will not die. So that's good. But your target is literally the heart. So that was the understanding of the time. And as science has evolved, the target became a little bit more well-defined. And eventually, for instance, it became clear that it's not just the heart, but it's a specific group of neurons on and in the heart that drive heart muscle to contract in a certain way, or perhaps neurons in your brain that control the heartbeat both directly and indirectly, because heart mostly controls its own heartbeat. And then later on, you discover that there may be receptors or ion channels on surface of these things. But again, that takes time. So the science has to advance.
So how did they find, for example, things like nitroglycerin? So this is like early 20th century? Or
yeah, this is probably late 19th century. I'll have to look this up again. Nitroglycerin was definitely an accident.
Nitroglycerin is an explosive. It's a popular explosive. How did they?
Why are you talking about
how did they? No, but that's a standard. That's a heart medicine.
Angina. It's a medicine for angina, which is
my grandmother would take it.
Generalized heart pain. And I think they might have discovered it because manufacturing workers were experiencing severe headaches and drops in blood pressure.
So they were basically poisoning the people manufacturing it. Were getting poisoned. Yeah.
So if you're in proximity to nitroglycerin, if you're covered with it, it'll go through your skin. And I think that they've noticed stuff like that. A lot of discoveries were done this way.
Why don't we see more accidents like this now? I feel like a lot of early drugs were discovered by accident.
So keep in mind...
Why are we not having these accidents anymore?
Because most of these accidents are bad. So most of the time people just die. So modern society, and the reason we live longer and we are generally happier, healthier, and fatter than we were before, owes a lot to not just medicine, but actually just sanitary and hygienic and safety practices that we've developed over the years. So it was acceptable to inject people with weird stuff back in the 1800s and even 1900s. You could get a complaint from a parent, and essentially your gravitas as a doctor would be like, no, trust me, I am a doctor. And then you inject their kid with the purely refined swine pancreas extract. That takes guts on both the patient, the parent, and the doctor's side. That does not happen that much anymore, or ever. It shouldn't really happen ever, because we've developed guidelines for ethical, safe testing of new things. To some extent, and I'm being a contrarian here, to some extent these guidelines also create barriers. So it is definitely easier to discover things if you're not concerned with ethics, but that's not how we should be doing things nevertheless. So causing suffering on the way, this is not, in my opinion, an acceptable way.
So basically, there were a lot of accidents because there were a lot of negative accidents, and some of them would be positive.
Some of them were legal issues, like no safety policies.
Yeah, no lawsuits. And then there were also known kind of like willow bark. People knew about willow bark, so then it was on the kind of laying there for somebody to extract to purify it. So we ran out of those things like all the typical kind of like remedies that were known.
Well, we still have that. And this is what's called the chemotype selective activities. So when you're discovering drugs, you can definitely discover them against the target if you know one. That's nice. You can discover them phenotypically using a cell line or some kind of an artificial system, but you don't know what the target is. And then there's a branch of that which is chemotype driven. So essentially what you're saying is, I know aspirin works. I'm going to take a whole bunch of substances like aspirin, and I'm going to apply them to a model system where aspirin works, and I am going to see which one of them gets the effect. So it's not quite random, and it's not quite targeted, but it's somewhere in between. But the important bit is chemotype. So you follow the chemical structure of a molecule as your guidepost. So you don't randomly screen. This would be, in modern times, this is somewhat referred to as targeted high-throughput screening. But again, high-throughput screening typically involves a target these days. So let's not go there yet. So again, discovery of medicine in the old days had a lot to do with basically either good luck, like the discovery of penicillin, because the mold essentially contaminated some petri dishes with bacteria, and it was noticed that bacteria were dying. It was relatively reckless, or after you receive the Nobel Prize, you can call it bold and pioneering. And at the time, they would be saying reckless and dangerous, sometimes even criminal, work with dogs and so on. The discovery of insulin, for example, or rather the commercialization of the insulin, tremendous amount of drama. And now it's easy to read about it, but it's become a little bit sanitized. But there were 4 people, Bunting, Best, MacLeod, and Collip, involved. Only 2 of them received Nobel Prizes. The other 2 received basically half of a Nobel Prize from the other 2 because they realized how important it was. They were fighting for dominance in the scientific arena. There was a lot of emotions. One of them beat up the other one because the other one wouldn't tell. So Collip, for example, refused to tell, I believe it was Bunting, how to purify insulin sufficiently so you can inject it without side effects. The secret was ethanol precipitation. So there was a lot of drama. And at the time, if you were living in 1923 or 1924 when this was happening, you would have almost felt like there's some kind of nefarious activity being done. And certainly people were accused of that. But then in the end, it ended up with the first ever Nobel Prize for Canada. And it made it on a $100 bill. So the vial of insulin and a picture, I believe, of Bunting is on it. So the point being is that a significant amount of dangerous experimentation was involved. And the big transition, and again, we have to thank, among other people, Claude Barnard, the big transition was experimentation on animals. People realized that animals and people are built on sort of general principles, and we share a lot of our build with even amphibians, so you can experiment. Certain things you could be doing on frogs, and certainly frogs don't complain as much, although it's still kind of mean to dissect them and so on, but better than experimenting on prisoners. For example, just to give you a story, in the sense that smallpox vaccine, everyone remembers Edward Jenner, who was a great guy. 1757, he was 8 years old when he received what was called variolation. So what was variolation? For that, we have to go back to 1670. People noticed that if you are exposed to smallpox by scratch of a smallpox-infected person, you have a lower odds of dying. You may develop smallpox, but it is weak. So you're infected, but in a controlled through skin manner. Somewhere around 1670, it probably started earlier than that, but the records at least are pretty clear. The Ottoman Empire was doing variolation. And so what it involved was taking a person who's sick with smallpox, puncturing their boil, right, puncturing the skin pustule, absorbing some of that stuff on a sharp tool like a needle, and then poking or scratching a healthy person, and then watching what happens. And what happens is the healthy person most likely will develop mild symptoms of smallpox. About 2% of the time, the person will die, unfortunately.
So that was the first vaccine?
They didn't call it vaccine. Society? Yeah, we're getting there. So it wasn't called vaccination yet. It was called variolation, or because vaccine has a specific origin. So in 1714, British Royal Society received, in 1716, received 2 letters, one from Emanuele Timoni and one from Giacomo Pillarino, that were describing these processes and how effective they were at reducing the mortality. Incidentally, a fun fact, some of the people who were variolated in Ottoman Empire were in fact in the harem of the Turkish Sultan. And it was done because smallpox is severely disfiguring. However, if you can control the entry point, you can make sure that the lesions that are left will be at a part of anatomy that is not too interesting. So somewhere on your leg, perhaps on your butt, as opposed to your face, which is where most of these things actually occur in the natural transmission. So your face gets ravaged by smallpox. Anyway, the British, however, were conservative. So British were not interested in these heathen practices at the time. They were like, nah, this is pretty barbaric and brutal. And it took a noblewoman, so her name was Lady Wortley Montagu, who lost her brother, and I think father, but definitely brother, and in 1715, her husband was appointed an ambassador to what was at the time called the Sublime Porte, which is basically the Turkish government, the Ottoman government. They were introduced to variolation in 1715, and she inoculates her 5-year-old son, Charles, who then inoculates her 4-year-old daughter, and both lived. So in 1921, under the supervision of the Royal College of Physicians, 6 prisoners were given variolation, and they survived. They were later infected with smallpox deliberately, and they shrugged it off, followed by an experiment on orphaned kids. So again, 1700s, people were kind of property, even in England, so it was acceptable. Then it avalanched from there. 2 of the daughters of Princess of Wales were inoculated, Empress Marie Theresa of Austria, Frederick II of Prussia, Louis XVI of France, Queen Catherine II of Russia. All of these were inoculated, all of them lived. And so in 1721, the practice of variolation was introduced in the colonies, so to speak, so America. Boston epidemic of 1721, there were 2 prominent doctors, Maher and Boylston. Maher convinced Boylston to do this, and for his trouble, his apartment got bombed. So people were absolutely pissed that this guy was spreading horrible disease deliberately. But this is also the very first time where medical statistics was used to defend a case like this, because they collected data on roughly 50% of Boston population was infected. So Boston at the time was 12,000 people, so 6,000 of them were infected. And 15% of those who were not variolated died, 2% who were variolated lived. And so that was one of the very, very early cases, if not the earliest case of using medical
15% of the uninoculated people died in Boston of smallpox.
Yep.
The people
and 2% who did get variolated did not. The only 2% died.
Only
So
2%. Yeah, I think you had it.
It's a tenfold gain in life. So you have a tenfold likelier outcome. And again, when everyone around you is sick, you start wondering, is this a good thing to do? And then again, 1757, so 30 years later, this is when Jenner, who really did a number on vaccines, was 8 years old, and he was variolated. He became sick, he recovered, and then he was safe. So we turn to Jenner, because Jenner was the guy who actually gave us the word vaccines. And that started by observing, which again, people have noticed before him, that milkmaids who milk cows, they get pustules on their hands from a cow version of smallpox. So a related virus called vaccinia, because vaca is cow, and that's how we get the word vaccine. Because instead of taking sick people, puncturing their pustules and smearing their stuff into the skin, you could do the same with cows. And the success of that technique is so much better. So you don't get smallpox. Your odds of dying are very, very small at this point, far lower than 2%. But your odds of not being sick with smallpox are excellent, like 98% or so. He proved his first experiment was on a kid called James Phipps. He wrote a communique in 1797 to the Royal Society, followed by a self-published book. It was kind of an interesting, rambling book about all kinds of things. But 1798, he published a book, and then he contacted prominent London physicians, Henry, Klein, Pearson, and Woodville. So basically, he gave the vaccination out. He was making no profit from it. He actually was very poor. The Royal Society eventually recognized it and gave him quite a lot of money for the time. But he was a truly selfless guy. He did not make a lot of money on it. And vaccination took off in around 1800s. And so it was so effective that European countries followed suit. Waterhouse brought it to Thomas Jefferson in America, who became interested in this, and America started getting vaccinated. And so in 1840, there was no more variolation. So you see this period, it took quite a while to go from just dying from smallpox to variolation, which reduced your odds of dying to about 2%, to vaccination, which reduced your odds of dying substantially lower than that. Sadly, the wife and children died of TB. Essentially, he was responsible for curing a very deadly disease, but his family perished from the one that we still have trouble with. So TB is still very, very bad. People die from it every time. Not every time, but a lot, especially in underdeveloped countries. So that's a story of vaccination, of how it all started. And you can see even though we like to assign, as people, we like to assign individual actors to things, and these actors are certainly clearly important and deserve recognition and all the prizes and so on. The reality of the situation is that it's the progress of science overall and the victory of rational over superstition, rational over preconceived notions. That is what makes it possible for us to do these things that we can do now with medicine, because the inertia of humankind is still very strong. Just look at recent anti-vax movements and all of this stuff with COVID.
Did they also look at that time into like side effects of the vaccines? Because, for example, after COVID, there have been published like a lot of cases when people have suffered from autoimmune diseases after that or something like that. I don't know if people have noticed at that time, or they were not really interested in those autoimmune diseases.
The biggest problem with variolation was that you could catch syphilis, because you're poking one person, right? You're taking their bodily fluids and injecting them into another person. What do you think will happen, right? If that person has the flu, you're going to get the flu. If that person has TB, you might get TB. If a person has syphilis, you're going to get syphilis. And that was one of the good things about vaccination. Cows don't really get sick with the same things we do. So catching something from a cow is a lot more difficult. It's not impossible, but it's a lot more difficult than catching something from a fellow human. So the process by which variolation became vaccination and the benefits that were derived from that were not just because the disease was milder. So cowpox is indeed a lot milder than peoplepox, but also because you did not get as much of the additional burden. I'm not going to dwell on the vaccine for COVID because it's a really hairy subject and there's still a lot of research being done. But it's suffice to say that from Ibn or Avicenna, right, everything you put in your body can be a poison, perhaps with the exception of sand because it's not soluble, and therefore you will die from obstructive gastrointestinal problems, but it will not poison you in the strictest sense. But everything else, like you can die with salt, you drink a gallon of brine, no, you're not going to make it. You might be saved in a hospital. The point being is everything is a dose. And Avicenna wrote that dose makes the poison. Many cases like that exist. And there is really not a lot of things that you can take that would not cause side effects. And those that you take that don't have known side effects simply means that we just don't know. But the benefit to risk ratio is what should drive individual choices. If you are going to perish in a year from uncontrolled diabetes, and then you have to go and inject yourself several times a day with partially purified insulin from a pig, you may develop immune problems, right? Because you're injecting yourself with a partially purified pig guts. That was acceptable to the people at the time because you get several years of life, decades perhaps. And the good news is as medicine progresses, these decades will turn into more decades. The classic case is with HIV. Early HIV drugs gave you a few years of life. But during that period, more drugs were discovered. And so you get another lease on life and so on. And nowadays, if you have HIV and you live in a country that cares about you, you will live almost a normal life without symptoms, without too much trouble. Used to be an absolutely death sentence. Used to be you have HIV, you're dead. But not anymore. Used to be you have type 1 diabetes, you were dead. Not anymore.
Just to kind of finish the history arc, we talked a lot about what drug development looked like before what you call like modern drug development. What about, let's say, 50 years ago, like early modern versus today? What happened in the early modern period and what's happening today?
So to go a little bit further back than 50 years ago, but 50 is okay. Some of the more systematic attempts at discovering drugs have to be credited to people like Ehrlich, who were fighting syphilis and more or less succeeded. And they discovered that you can administer to a test animal, so rabbits are susceptible to syphilis, you can induce syphilis in them, and then you can observe the progression of the disease. So you can take a bunch of rabbits and you can treat them with substances off the shelf, essentially, until you find, if you're lucky, you find one, and so for him it was 600 and something, 606, I think, where the rabbit cures. And so it was salvarsan, an arsenic compound that preferentially kills off syphilis and doesn't kill the rabbit or people that much. And again, remember, arsenic compound, you take enough of it, it's going to kill you. The trick is don't take too much. Same with insulin, incidentally. Inject yourself with enough insulin, you're going to go into coma and die. The point is the system has become the norm. What I mean by that is you do your best to discover what is the causative reason for the disease. So lack of insulin, for example, diabetes type 1, incorrect function of your blood pressure regulatory system, lack of certain hormones like the human growth hormone causes dwarfism, and very sad. And then you go and you find substances that will ameliorate that problem, either in a modern or the not so modern case. So what changed with time is the advancement of molecular biology, advancement of chemistry, and it's in reverse order. Chemistry was first, and general understanding of biology and physiology. So people started to be able to isolate in a completely artificial system the molecular reason for a certain thing. So, for example, let's take a cytokine. A cytokine is a protein, small protein, that your cells secrete, and then either those exact same cells or other cells absorb, and they react to a cytokine with either anti-inflammatory or pro-inflammatory response. Depends on the cytokine. So if I want to do something with the cytokine, I can isolate it now. I can make it in a bacterium or yeast or cultured human cell, and then I can expose that cytokine to a variety of chemical substances, and I can observe these substances interact with the cytokine. At this point, I don't know if they will do something on a cellular or organismal scale. I just know that the vast majority of my substances, literally 99.99%, do nothing, or they just stick to it in some non-specific way. But that 0.01%, I'm now going to take further. Now I'm going to take the cytokine and I'm going to treat a cell culture, and basically I expect a response. So if I treat a certain cell culture with my recombinant, meaning artificially produced cytokine, I expect those cells to start secreting a second cytokine. So for example, IL-17, I treat my cells, I expect IL-6 to be secreted. If I add a substance into that experiment and my cells do not secrete IL-6, then I've somehow interrupted the signal by IL-17. And that's my next step. So it's still quite a model. It's not even a mouse. But now I know that I have a substance in my hand that at certain concentration essentially arrests the action of the signal. Pretty cool. How far is this from a drug? Very far. So this is still hit finding, hit validation stage of what's now considered a drug discovery paradigm. So you begin by finding these substances that do something interesting. That's something you define as interesting. Then you take these substances and you do in parallel a bit of optimization in the chemical space. So you make them more potent. You also want to make them less susceptible to degradation by human body. So excretion and degradation, transformation.
How do you do that? You mean you adjust the chemical structure?
Yep.
And is that random, or it's kind of driven by past known good chemicals, or?
It's a very tough thing to do to begin with. But it can be both random and deliberate. So Pharma has accumulated a vast body of knowledge, essentially what parts of a molecule are, for example, subject to chemical attack by your body. So the human body or any other animal is a machine designed for survival. So we certainly ingest a bunch of poisons every day. Like you eat a burnt toast, you ingest a whole bunch of poisons. You don't know it because you like the smell and the taste. But actually nor the smell, neither the taste nor the smell are good. Those are chemicals that if you drink like a gallon of them or even a cup of them, you will perish. But your body can deal with small quantities because we have enzymes that attack these molecules and convert them into harmless ones. We have enzymes that conjugate the harmful molecules to a message that says, kidneys, take me out, and then it gets peed out immediately or pooped out or sweated out. Either way, we have ways to deal with bad actors. But sometimes or oftentimes the drug that we're trying to administer is perceived by your body as one of those bad actors. And so there have to be adjustments made to prevent that from happening. And so roughly 60 or 70% of drug discovery post-hit finding has to do with what's called as DMPK, so distribution, metabolism, pharmacokinetics type of stuff. How much of the drug that I take as a pill will make it to my bloodstream? How much of it will survive liver metabolism? How much of it will survive additional metabolism and excretion? How much of it will then get reabsorbed by the cells that need to reabsorb it? What will happen to it in the cells? Will it bind to some random proteins, or will it bind to albumin in your blood? And is it a good thing or a bad thing? And sometimes it can be either. Sometimes binding to albumin is good. Sometimes it means that it will circulate longer. Sometimes binding to albumin is bad because it will.
So just a note for
stuck.
non-biologists, albumin is it's called the carrier protein of your blood. So it's basically your blood has a really high percentage of this one protein that kind of sticks to a lot of things a little bit. So all the small molecules in your blood, like hormones and things like that, a lot of them are not soluble in water. So the way that they're dissolved in the blood is that they're actually partially stuck to this protein. And there's a massive, so if you look at blood, the top ingredient is this protein. That's kind of why serum, if you get rid of the cells, it's like a thick yellow substance. A lot of it is this protein albumin.
So then, like, from the chemical perspective, let's say for the drug, would it be fair to say that it's essentially for the drug discovery, try to find a molecule which would do something in the body, and then what are the other proteins or molecules are available there, and then that's the way where you try to target it. Let's say that molecule need to do something or make the cell to produce the specific proteins.
It's a multi-stage process. Initially, you need to find a molecule, a chemical molecule that attracts you because it has a salient feature. It produces an effect in your model system that is desirable. A model system could be as simple as a single enzyme, right? It could be as complicated as a chain of enzymes, or it could be a cell or cell culture. It could be an organ culture, so stratified group of cells that create, for example, a blood vessel.
Just to be clear, what Artem is saying is literally you put the chemical into a tube. The tube has essentially water with some salts and so on, and let's say an enzyme, so one protein. Function. That protein's supposed to break something up or build something, and then let's say you want to stop that, right? So you put a chemical in the tube and you see if the reaction stops, and then it's like that's a hit. So
that's a hit. Like, and then again, I'm trying to be as detailed, but perhaps too detailed. The hit can be an invalid hit. For example, if you take soap, just general soap, and you add it to almost any protein. It's not entirely true, but I'm simplifying. If you take soap and you add it to your enzyme, very good chances are your enzyme will die. And so you will have a hit in a screen.
If your goal is to turn off that enzyme.
The problem is it will kill any other protein also. So it's not specific. Yes, it's effective, but it's no more effective than curing epilepsy by killing the patient, right? So that's not good. You've cured, certainly the patient didn't make it, but the epilepsy is gone too. That's great. You have to find a substance that specifically addresses the thing that you want to address, be it a protein, be it a cell, be it a piece of DNA, be it an interaction between two proteins. That's what you're trying to do. And so at that point, when you become convinced that you have a small molecule in your hands, or an antibody or a peptide and so on, that addresses something rather specifically, then you can go and begin its long journey to a drug. And that long journey will encounter many pitfalls, and most things don't make it, which is why you never start with one. You start with a set of these things, dozens preferably.
And can you do something like inverse engineering? So you have a target process, and then on top of that, you put the conditions that it shouldn't initiate the other processes.
Right, and I have to remember I'm speaking with physicists, so it's a very good analogy, but let me transform it into biological terms. If you have, let's say, a disease state that is characterized by a broken protein, so your protein just doesn't work as it's supposed to because perhaps the gene for it is malfunctioning, is corrupt, or even missing, a very attractive option is to reintroduce the healthy version of this protein back into the human body. And you can do it in a number of ways, but basically, if it's a protein that just happens to float in your blood, quote unquote, then you can inject it and then hope for the best. And there are certainly medicines like that. Or if it's a more complicated case, like a protein is intracellular and also it's ubiquitous, so it's in every cell of your body, then you have a lot less options available. So basically, this is bordering into gene therapy, right? So you have to introduce a gene into a person and attempt to replace or supplement the broken gene. And there are approved gene therapies out there. They can be probably counted on the fingers of two hands at this point, maybe even one hand. And it's certainly not possible yet to introduce a gene into kind of every cell. It's possible to introduce a gene into populations of cells and have it function reasonably well. This is where the very exciting future lies. And the difficult part is that in a body, a human or animal, everything is connected. It's truly a holographic or holistic system. It's very difficult to change something and just have one outcome. And our tools, as much as we love them, are still quite crude. Even though we have now adenoviruses, lentiviruses, we have CRISPR, and we have other techniques, but still, these are still very early. We cannot target individual groups of cells with a good degree of selectivity. And even replacing genes, we still have to be careful because we can replace things that we didn't intend to replace. So there are complications. And this is why I think one of the reasons why this is so hard is because we still understand only a fraction of the total, even when it goes just one cell. Certainly an organ or a whole organism, we understand very little. That is not to say that we don't understand. We understand a lot, but in terms of whole, it's a small fraction. There's a group of people, I think, that tend to say human bodies are so complex that a person is unlikely to understand them, and therefore the job of understanding is relegated to a computer. I think we will find out, because a computer doesn't have a limitation on how much it can remember and hold in one memory, but we haven't seen that really happening quite yet. But it is an interesting question. Can a person, in fact, understand, without abstraction or reductionism, all the processes that occur in one cell? And it's tempting to say no. So we're clever, but are we that clever? Because most of what we've accomplished so far, 99% lion's share, has been done through tried and true principles of abstraction and reduction. So we either create a model of something that we understand, like saying that insulin is like a little guy who runs and turns on a valve, and then glucose pours into the cells, and the cells metabolize it into interesting things and store it as fat, store it as glycogen. But there is no guy, okay? There is no valve. It's all much more complicated. Or reductionism, which is to say, okay, I know that insulin binds to insulin receptors, and then these get internalized, and then they throw off a cascade. That is just one line. Even if it has little loops and branches, it's still just one line in a drawing that has millions and millions of lines. So we've reduced that entire painting to one line, and we forget essentially all the secondary effects. Because insulin, for example, is also involved in cell proliferation, as it turns out. So is it true that you can just give it to people and everything will be great? Practice says mostly great, but diabetics are never quite healthy. Even with the best care, they're still suffering from things. And it probably has to do with the fact that their systems are disrupted in ways that we understand only partially. The holistic picture is difficult. And this is not to say, again, it's not to say that we don't know something and we are just plunging blindly ahead. No, it's a risk and reward balance. The person will die if you don't give them insulin. Of course you should. But you also should understand what happens to them on a deeper level as time goes on. So essentially don't stop at insulin. And now we have second and third generation things like the semaglutide and so on, which attempt to control the system with a more understanding. So there are multiple hormones involved, right? So insulin was the first one and glucagon was the close second. But now we know GLP-1, GIP-1, GCG, and so on. So
these are the Ozempic weight loss.
Right,
peptide.
so Ozempic, so specifically Ozempic is one, right? Tirzepatide is two, and there's now a third one called retaglutide, which targets 3 receptors. So it targets also glucagon receptors on top of the GLP.
What's a receptor? Let's start with like a basic.
Receptor is that mystical tap or a switch that tells the cell that something interesting has happened. So if you think of a cell, right, it's basically a plastic bag. It's a jelly-filled plastic bag. It's surrounded by a relatively impermeable membrane, and there's all kinds of interesting things both inside and outside. And so the whole thing about a cell is that it's a region of space that is bounded by a specific thing, like a plastic bag, tight, right? So how does a plastic bag talk to the outside world? Well, it has to create holes and openings through which information can pass and materials can pass, signals can enter and leave, and so on. And some of these holes are channels, so they are mass drivers, if you will. Some channels are also sensors, not all, but many. And then there are things that are considered to be specific for sensing, and these are receptors. And there are many different types of receptors. In this particular case, this is G protein coupled receptors, which are a certain type of receptor that is very, very popular. It's an ancient form of sensing. And what they do is they receive a stimulus from the outside in the form of a let's say a peptide or a small molecule.
So peptide is like a very small protein, basically.
String of amino acids. So everything we are is built of certain building blocks.
Here's an interesting question, an interesting almost physics question that's very related to this. Why is insulin a peptide, not a small molecule? What's the reason that some hormones are small molecules and some hormones are peptide? Is there a functional physics reason? Is it that it has more binding sites, or?
That's a great question, and I wish I could answer it, because that means I would understand a lot more than I do right now. In a nutshell, I think it has to do with whether their effect is local or global, and whether their effect is severely or less severely time limited. So if you want to limit something in time, like you send a message and then you want to stop, essentially that message has to self-terminate. It's relatively easy to do that if the message is peptide, because
Why?
peptides decay. They get eaten by proteases. They decay spontaneously also, but they get eaten by proteases. They decompose them into chunks, ultimately down to individual amino acids. Whereas a small molecule, if you're not careful, it can persist for a while. And so certain hormones are better off as small molecules, others are better off as peptides. Why exactly our bodies decided to do that, I don't know. Furthermore, I wouldn't be surprised, I can't name any examples offhand, but I wouldn't be surprised if there are systems in our distant cousins, like protists or something, where signaling molecules that are, for example, there are peptides in us, are small molecules in them, right? So it may be that a small multicellular organism, like a coral polyp, only needs to signal, so much that it can do it with a small molecule, whereas a bigger organism like us needs a big old peptide to do that. It's not clear that this is the case always, but I can tell you that it has a lot to do with the way these molecules are distributed and the way that they're degraded.
If you look at the information storage, if you have a small molecule, there's only so much information on that molecule in a way, whereas a peptide has more sites, right? It's a bigger thing.
That's entirely
possible.
possible.
Is there something related to, like, it can be more specific or have more information, not just one site, but maybe it has secondary sites that give other
That's entirely
functionality.
possible. And certainly there's many examples. So you're 100% correct in that aspect. I think that you're completely right that a bigger molecule can do more, essentially. It's not always the case. So one of the most potent signaling molecules in our body is glucose. This is pretty simple, 6 carbons, nothing much to it. And it orchestrates an incredible variety of things. So the information can be encoded in the molecule itself, yes. But it also is encoded in where it is at any given time and what is listening. So you can encode information. Essentially, you can throw a book out of a window, and if you tell the person down there, hey, I'm going to throw Tom Sawyer, that means run away, you've been made. Versus if I'm throwing Anna Karenina, everything is cool, you can proceed with robbing the bank. So you're throwing a bunch of information out there, but the meaning is essentially like one byte. Glucose is the case where a staggering variety of things depend on knowing how much and where it is, and yet it is itself very small. But yes, you're absolutely right. It has to do with information content.
Right, so in the case of glucose, it's like an amplitude signal, because it's really about how much there is.
For some cells, but for other cells, it's also where it is, right? So for instance, to give you some food for thought, we and cows, we're not that far, right? Smallpox, cowpox, great. But cows rely in their metabolism much more on simple fatty acids that are 3-4 carbons long, butyric, propionic, than they do on glucose. I mean, of course, they're still mammals. They still use glucose in a variety of critical cellular processes, but a lot of their energy is derived from grass via essentially complex bacterial and protozoal fermentation of grass in their multitude of stomachs. And the first thing that they receive from these processes are, in fact, fatty acids. And so a cow has adapted its metabolism to depend on that. And so for it, glucose has slightly different meaning than, for example, for us.
But I think overall also it depends on type of the processes, because from my side, I just started recently to study like neuroscience, and then for neurons, it says that you can activate the channels by ions or by these neurotransmitters, which could be, as you said, like some types of acids or
stepping
components.
all the way back. If you look at, again, a cell, this little plastic bag that is alive somehow, which is, let's not go there, the definition of life is giving me nightmares. What can it sense? Okay, well, we know certain cells can sense electromagnetic radiation, typically in the form of light. They also can sense heat, which is both electromagnetic radiation and motion of molecules. They can sense pressure, so pressure sensors. They can sense stretchy forces, stretchy or compressive forces. They can sense any number of chemical stimuli. So certainly as simple, the simplest chemical stimulus that I can think of is a proton, H+. It's not really H+, it's typically H3O+, but you know what I'm saying. Something is acidic or basic, right? More protons floating around or less. That's as simple as it gets, really, in chemistry. And they can sense very complex signals like part of a bacterial cell wall. Our entire body is permeated by cells who do just basically one job. They sit and they listen whether there is a piece of broken bacterium floating by. And the moment they sense it, they're like, uh-oh, that means I've been infected and I need to trigger an alert. So they're toll-like receptors that are just sitting there sensing pieces of broken bacteria, pieces of viruses, pieces of fungi. And the reason for that is that over millennia and millions of years, life has adapted to be preyed upon and to resist predation by bacteria, viruses, and fungi. So we carry with us all that baggage. That's pretty awesome. So the signal can be anything, but certainly when we feel the pressure, one of the receptors that we don't have is hygro receptor, but insects do. So we cannot, you can touch a drop of water and you can deduce that it's water, but you can be tricked. I can take a drop of silicon, cool it down a bit, and make it just right. So you'll touch it and you'll feel watery, but it's not water. Whereas you cannot fool an insect. They will touch it and they'll be like, nope, that's not water. I have water receptors on my legs. I know what water is. We don't. We infer. So we can drink horrible things and think they're water, but they're not. Bees do not sense sugar as well as we do. We sense sugar about 60-fold better than a bee. Why? Because they live on flowers, they drink nectar. If the nectar is inferior, they don't want to drink it. It's a waste for them. They want concentrated sugar to bring home to make honey. So they don't even taste things that are like not syrup. Whereas for us, syrup, like holy cow, I don't put too much of it in my tea. So the point being is that the one thing that I think is the hardest for cells to sense, and they still can, is just really hard, is magnetism. And there are some very wild theories out there, much more in your realm than mine, how certain cells can sense magnetism. Some of them involve even entanglement between particles, believe it or not. I don't know how well this is proven, but there's cutting-edge stuff. Others basically are more sort of, I wouldn't call it pedestrian, but more basic. Basically micromagnets that turn and twist. Cells can sense gravity, by the way. Plants, right? You take a plant, you plant it, and it knows where to put the root and where to put the leaves. How does it do that? It's obvious to us. Well, it's like, look up. The plant is dumb. It doesn't have that. So it knows because there's starch granules in the cells of a plant that settle, and they exert pressure on the side of the cell that is pointing down. And a plant knows. And it senses gravity. There's beautiful experiments in centrifuges and stuff showing that, yes, it is in fact gravity. You can trick them with light, but they will know where to grow the roots. And the roots will grow in gravitational direction by basically growing a plant on a centrifuge. So you can make it grow sideways because it knows where the gravity is. Or in this case, it's a force, fake gravity, right? But still, it knows. Almost everything can be sensed. Difficult ones are magnetism. Electromagnetic radiation in long wavelengths is hard. In fact, one of the things that I would pose to our listeners and you guys as a question is, how can a living system develop an antenna? What can be done, right? How do I become listening to like microwaves and stuff? How do I do that? What do I need to grow in my cell? Because the cell is kind of small, so what portion of a wavelength can I use? Inside one cell is not a lot, right? There's so many microns, and radio waves are kind of long. So I'm now talking about 1,028th of a wavelength, not a lot of signal there. So how do I do that? How do I listen to electromagnetic radiation if I'm alive without using metals and semiconductors? I don't know. That'd be cool.
That's an interesting question. A lot of people think they can feel when they're next to a radio tower and things like that, but
just next to even the microwave.
Or like a cell phone.
You can probably feel the heat. Birds do fry in microwave radars. Like one of the reasons that microwaves were discovered for food is that a guy had a chocolate bar in his pocket in foil and it melted. So he was like, oh, I can cook with this. And then microwave was born. But that was a military research into microwave radar.
I want to shift discussion a little bit. Since you mentioned Ozempic, and I think many people would like to know more about it because it's all over the news right now. We all know its ability to help people lose weight, but you mentioned it in the context of diabetes. Does it really help to treat diabetes, or how Ozempic works in reality?
That's a good question, and specifically Ozempic is definitely better for diabetes than for weight loss. It was developed for diabetes, and then during the process of its testing, it was a beneficial side effect, that weight loss. Since then, a lot more has been done in other medicines that do actually more for weight loss than Ozempic does. So very, very quickly, going back to 1845, our friend Claude Bernard discovered this whole deal with glucose, where you eat it and you can tolerate a lot, you inject it and you cannot tolerate a lot. So right around that time, people started speculating that there are hormones, additional hormones beyond insulin and glucagon, that are responsible for this effect. And Jean dubbed it incretin. And it basically was assumed to exist before it was actually found. And incretin is one or more hormones that are secreted by the intestine to aid in insulin secretion. And also, as it turns out, incretin are responsible for slowing down digestion. So your stomach contains your digestive juices and food, and so it parcels about 7 to 15 milliliters at a time into the intestine. There is a sphincter that opens and closes, and incretin controls the rate of opening and closing of that sphincter. And by the way, the reason why such small portions are released is because stomach is extremely acidic, and your intestine is actually fairly basic. But that balance is easy to shift. So if you release all at once, it basically will burn the intestine with acid. Intestine is alkalinized using bicarbonate, which incidentally also is secreted from the pancreas, because the pancreas is a dual-purpose gland. It secretes hormones into your blood, and it secretes gastric juice into your intestine. It contains digestive enzymes and bicarbonate. Anyway, somewhere between 1932 and 1960, people have started pointing out that there are all kinds of hormones, but still it was difficult to isolate them pure, that definitely stimulate insulin production when you eat. Right around 1980 is where things get really interesting. And things get really interesting from two alternative directions, and they both involve weird animals. One weird animal is the deep-sea anglerfish. So in 1982, a group of people, including people like Joel Haberner, Shields, and Lund, were collecting deep-sea anglerfish from fishermen. So they would go on boats, and during the normal fishing process, you sometimes pick up these anglerfish. They're hideously ugly, like weird little creatures. And in them, the cells that produce glucagon are easily identifiable. So they're easy to harvest. They would freeze them on dry ice and then dissect them later. Anyway, the point being is that the deep-sea anglerfish glucagon opened the door to studies of incretin, because incretin peptides are encoded essentially downstream in that organism. So you can find them real easily. And the studies with...
Sorry, what do you mean they're encoded downstream? You have to unpack that.
If you consider DNA to be like the most basic analogy is your ticker tape that encodes instructions for things. So there are codes for glucagon and codes for incretin peptides. They're in close proximity to one another. They're basically right next to each other, which in general in organisms, that type of organization suggests functional involvement. It's not always the case, but often is. So if your instruction for making glucagon and some other peptide are close together, physically close on DNA, it's not a difficult assumption that other peptide is involved. And so they found glucagon and these GIP and GLP peptides in anglerfish in close proximity, encoded in tandem. So anyway, the one side, this opens the door for glucagon to be a therapeutic, because glucagon is actually a therapeutic. It's prescribed for critical hypoglycemia. When you run out of sugar, and even if you take glucose, you're still low in sugar, you can inject glucagon and your body will release its own sugar from its stores, right? So it'll hydrolyze sugar polymers in your liver. One side of the story is deep-sea anglerfish, 1982. The other side of the story is equally weird. It's Gila monsters. You know these toxic lizards? NIDDK researchers went and started studying what Gila monsters, which can go for a really long time without eating, how do they regulate their sugar? And so they found a bunch of substances that really allow the Gila monsters to extend their metabolism for a long time. So they digest very slowly, they release sugar very slowly, but very steadily. And so they found these hormones that they called, among other things, extendin-4. They shopped them around, and the folks involved were John Pisano and John Eng. So they shopped them around, and interestingly, many pharma companies refused to pursue this direction. And the reason they refused is because they didn't want substances from toxic lizards to be in human pharmacopoeia. They were like not responsible. They were not going to be responsible for that, even though it's just silly. What does a substance from toxic lizard have to do with toxic lizard, really? Anyway, eventually a company named Amylin took this on and produced a drug in 2005 that they called Byetta.
And so the theory for why this was an interesting drug is that these lizards have I guess the idea is it's appetite suppressant because these lizards eat very rarely.
Among other things, yes, but it's more than appetite suppressant. Again, this
It's
is
about
diabetes.
sugar control.
This is
It's about
diabetes
glucose
drug.
control.
So what it did is it allowed people to regulate their insulin production. Rather than just injecting insulin, you could inject this stuff, and it would in turn regulate your insulin production and your glucose metabolism. So there and still there are things that we don't understand, but at the time they certainly were interested in seeing how it goes. But Byetta was approved in 2005, and what people noticed is that it caused about 6% weight loss in people, 5-6%. And so they started realizing that this was interesting. In parallel, Novo Nordisk produced this medicine called Victoza, which was a daily injectable, also based on these peptides, on GAP peptides. Daily injectable was not so good, because it's annoying to inject yourself every day. But then up to 2017, they worked on making it progressively more stable and easier to preserve in the human body, so to speak. And so in 2010, Victoza, which is liraglutide, transformed into Ozempic, which is semaglutide, which is a modified peptide. And so again, during the
So they took the original hormone and modified it to make it hard to break down.
Harder to break
in the
down,
body.
more prolonged mode of action. Because among other things, injection of these hormones in the early days, they would cause like uncontrolled vomiting, for example, horrible nausea. It wasn't so great. So over the process of development, this transformed into what we now know.
Let's say you're injecting people with this. They're having uncontrolled vomiting. What do you do? What's next? Like, what's your next guess? What's your next move? How do you connect this vomiting to the structure of or you know what I'm saying? Like,
among other things, I wasn't there doing it, but I would not be surprised if they did dose dependency studies. And they found out that, for example, for different people, the doses are slightly variable. So in this case, what you want to do is you want to slow the uptake. So you want to make sure that even if you inadvertently give a slightly higher dose than what you wanted, because you can't really dial it in for every individual person. We can't all be Jeff Bezos who can afford to do that. Right, so a physician of a very wealthy person can spend 6 months dialing to the T the exact regimen of something. So you, being the wealthy patron, are going to pay me to study you for a year and tell you that you don't need 0.25 milligrams of Ozempic. You need 0.17 milligrams unless you ran that day, and then you need 0.36. So you work out this precise, very, very detailed regimen, but it's certainly not for everyone, right? You can't sell it to millions of people. It's not going to work that way because you can't afford to spend the time.
Could you go deeply a little bit, like if I'm Jeff Bezos, what kind of procedures needs to be done to find the proper amounts of the medicine?
Yeah, what is the indicator?
It would basically begin low and you keep rising it until either the therapeutic effect is at its maximum or you start seeing side effects. So it's no different from what people do during clinical trials. The difference here is that it's a clinical trial for one person.
And how to understand that it's a peak of its performance?
So in this case, you, for example, want to make sure that your sugar level is completely stable. So you're supposed to have between 80 and 120 of your glucose mg per deciliter. And
so
deviations...
glucose monitor can help, is that right?
Well, for example, yes. And nowadays, people have a wearable glucose monitor, but early on, you could just take blood. So my point is that if someone has the resources to dedicate their well-being as their sort of top priority, not just for themselves, but for a whole team of people who are just going to be into that, that certainly is helpful, but it's not for everyone. And so, of course, pharmaceutical companies want to make a fairly democratic drug. That's the best way to make money. A democratic drug has to be prescribed in certain increments. There's 0.25, 0.5, 0.75, and so on, however the increments are. So my point is that the more you have the ability to control the uptake, the distribution and the decay of a particular medicine, the more you can dial it in such that a large percentage of population will receive the benefit of the medicine without the bad side effects.
So then it looks like if there would be at-home diagnostics widely available, then you can by yourself change the dose, or maybe you will discuss it with the doctor and find the right amounts.
If you have access to compounding pharmacy. So when you receive a dose of Ozempic, for most people, you receive it in a pre-filled syringe or a pre-dialed thing. So you don't have a control over that. You have moderate control over that. You can inject yourself once or twice, but it'll be increments. If you have a compounding pharmacy at your disposal, which you certainly can do, they can formulate the same exact medicine for you in a syringe where you have essentially analog control, not digital anymore. And you can inject yourself with 0.17 milligrams today and 0.38 milligrams the next week if you want, right? If it's necessary.
It's like a feedback loop, essentially.
If it's necessary, yes. But that feedback loop is not safe, right? So you have to be absolutely sure you know what you're doing, which is, again, where medical professionals have an edge because they are trained and ethically bound to do that right. And again, not everyone can afford to do it, although I'm perfectly happy. People should consider their body in its holistic sense, meaning they should consider what they put in and what they want to get out. But I wouldn't recommend tinkering. It's a little bit tricky because you're tinkering with basic functions of your physiology. If you cause yourself to go into hyperglycemic or hypoglycemic crisis, well, you could die or you can end up vegetabilized. Right? If your body doesn't receive enough.
I think the way we got into this topic is that you're saying that what they did to reduce side effects potentially, so your guess is to change the way that smooth the uptake such that the dose is less important. Basically, because there's kind of like a capacitance effect.
Yep, and it's actually the same with insulin. If you consider insulin, it's a lot easier to explain in the case of insulin because it's much more studied. Insulin is given to people in two forms, the fast-acting and the slow-acting. And it's typically given as a mixture of the two forms. And it's not a secret on how. Fast-acting insulin is actually soluble, so it's a solution, whereas slow-acting insulin is crystalline. It's a very precisely controlled microcrystalline product. And these crystals sit in your tissue and gradually dissolve. So slow-acting insulin gives you that slow but steady rate of insulin secretion that is supposed to, in a way, mimic how your pancreas would normally do it. And the fast-acting insulin is basically, okay, I just ate, I need a shot of insulin because I need to absorb all of that glucose and other nutrients. And so it accomplishes both. So with other hormone therapies, there are mechanisms similar to that, although it's not necessarily crystalline or not. But you can imagine some things, for example, going into your fat as a depot and then slowly diffusing out of the fat. That's a very common method.
And what's the trick? Let's say if you take the Ozempic or these related peptides, is it attaching like a fatty chain to the peptide?
Ozempic specifically has a fatty chain. It has a big fatty chain on it, which, again, I recommend people picking up their own literature, but I believe it has to do both with the half-life and the distribution and stability. So it has a lot to do with its pharmacokinetics in the body. The point on all of this is that Ozempic was, I speak for myself based on what I'm reading, is developed primarily as a diabetes medicine, but the fat-reducing side effect, it was very welcome and was noticed, again, before. So things like tirzepatide and what's currently in clinical trials, retaglutide, they are definitely looking at addressing the fat aspect of things. So it's not known, for example, that tirzepatide and definitely retaglutide, they actually increase turnover of fat. So they accelerate the way, so somehow, it's not entirely clear exactly how, because it's complicated, but they address fat burn, not just storage of glucose. It's a little bit of a paradox because glucagon, for example, is supposed to increase your glucose in the blood, right? And yet, when you use these peptides that, among other things, they can target glucagon receptors, that's not quite how it works. There is a short-term and a long-term effect, and these things are very finely balanced. I think that we are in an exciting period of the development of these medicines, and I think that they will be probably even better than they are now with time. And this is both good and sad at the same time. Good because, like many Americans, I am a little bit overweight, I can use that, and many Americans could use that. It's still not available to everyone because not everyone has the money to afford it. Not everyone has the ability to articulate to their physician the need, right? And so it's a little bit sad because it is definitely a first world problem, right? We're fat because we have all kinds of food to eat. We're fat because we have candy bars, right? Like this is not necessary for my survival, but it's highly, highly pleasant. Whereas people are still dying from tuberculosis and malaria and
so Artem, you grew up in Ukraine, right?
Mm-hmm.
What was your diet like when you lived in Ukraine? What did you eat as like a kid when you were 10 years old?
What we all ate. Those who can afford it had meat relatively frequently, and then vegetables, potatoes, soups, the regular.
Were you in the countryside or in the?
I was born in a city.
In the city, so you weren't like picking mushrooms.
I was not, although I was picking mushrooms, and I got poisoned by mushrooms at one point, and I since then don't eat them anymore. It created a psychological thing where I don't want to eat mushrooms because of not so fun experiences. And by the way, I wasn't poisoned by a toxic mushroom. I knew my mushrooms. I was poisoned by a mushroom that was growing too close to the road, and we had leaded fuel at the time, and mushrooms are notorious for accumulating heavy atoms. And so several of us ate mushrooms that were probably harvested close to the road. And so they were probably full of lead. And the symptoms that we've had were consistent with mild intoxication by heavy atoms like lead. But anyway, mushrooms are no for me anymore. Returning to Ozempic and so on, we will see an improvement in all of this. There's even an oral form available, but it's absorbed through the gut only at 1% of the total dose. So you have to take heroic amounts of it and adjuvants. I'm personally not too bothered by injections, especially since these injections are incredibly tiny.
On the other side, I'm like more into sports and fitness, and what my fitness instructor would say, he was like, it's all about the balance of how much carbs you put inside and then how much you use them. So essentially, he would say, if you want to lose the weight, you have to spend more than you get carbs. So you can, for example, cut on your carbs or maybe increase like the metabolics so that your body burns them much. So I feel like maybe in the United States, it's more like we're trying to find like a magic pill. You just take it.
But that's great, right? Magic pill. So America is this place where everything sucks, but because of that, people are like motivated to get these magic pills. Like, it's like you don't live next to your family, everything's depressing, everyone's fat, but that motivates you, so you do more new interesting things. It doesn't have a lot of the comforts of like ancestral existence. It doesn't have that, but it has a lot of new things to replace it.
So I'm all pro exercise. I love the idea of it, and I love the practice of it, but I can also sympathize. So there's two aspects that I want to address because you've touched on a very specific and very interesting topic. Firstly, physiology. So it appears that when a person exercises, somehow, after a while especially, his or her body becomes aware of the unusual caloric expenditure, and it's actually compensating during the rest of the day. When somebody who's not like a dedicated athlete, who doesn't spend 6 hours in a gym, they go to a gym twice a week or 3 times a week for a couple hours. The rest of that week and the rest of that day, your body is actually saving the energy. And so there's a considerable amount of resistance. And so that basically isn't enough. And that's assuming that a person has the strength of will and, more importantly, the free time. Now, consider a situation where someone is a single mom working in a fast food restaurant. When would they find the time or the energy or the money, for that matter, to go to a gym and spend 2 hours at a gym running or lifting weights? It's unlikely. So they're likely going to have to go and pick up their kid from an American horrible daycare that costs like a third of their salary. So I sympathize with people not because they are lazy and unmotivated, but because often people are unable to find the resources, the time to do what is right. They even want to do what's right, but they can't. So for those people, a magic pill is certainly a great solution. If you can give them 10 years more life, because they're not fat anymore and their sugar is under control and their toes are not falling off because of the late stage diabetes, go for it.
I actually think that there's an interesting effect that might happen from Ozempic, and especially if there's like next generation ones that are cheaper and even more effective, is that there might be like this latent immense productivity in America that'll get unlocked because like 60% of Americans are overweight or whatever. It's an enormous, I think maybe it's obese is 60%. I don't know the number, but basically if you suddenly solve this, there might be like an explosion of all sorts of great new things because like being obese, you're sick, this, that, it has many downstream effects. Whereas if they're all relatively healthy, I mean, America is already extremely productive, right? But if you then if you have everybody, if they're just, let's say, I mean, think of it from a really basic level, like if you on a unit cell level, right? If you take one person, if they're like, let's say they're sitting in a chair and there's a productivity gain that's possible if they get up, go somewhere, get something and sit back down. If they're fat, they're just less likely to do that. So they're going to instead send an email. Instead of going and talking to their colleague in the next room, they'll send him like a Slack message and then nothing will happen.
Now it's everything moving to digital kind of work online.
Yeah, but
So
I think, part of that could actually, I mean, that's what I'm saying. Maybe there's a lot of productivity gain in just people having more like nervous energy. People study this, and a lot of the really prolific inventors and whatever actually just didn't eat much at all. So like they're just known for eating very rarely because they're so focused, on what they're doing, so excited, they forget to eat, right? So they're not like during especially the most productive periods of their life. And so if you suddenly give everyone a drug that makes them feel that way, it takes away their appetite. And another very interesting thing that I've seen is that Ozempic, there's like Reddit threads like r/Ozempic where people talk about like they had a gambling addiction and then they started taking Ozempic and it went away. So there's something like really deep about what it's doing where it's like removing cravings. Maybe there are some crazy side effects that we're going to find out about, which is also interesting. Like I wonder what's going to happen in the next 5-10 years. But there is this possible accelerationist interpretation of this where you just give this to everybody and the whole country blooms until suddenly you get like Silicon Valley in Kansas, right? Where you have like 80% obesity. It goes 5% and you get massive productivity gains or whatever. So I don't know. That's like an exciting future, right? Where pharmaceuticals do something not just bring you back to normal, but even maybe, incredible, right? Because when people were skinny, let's say in the 60s and 70s, they didn't have the internet. Now we have all these things, right? So if we get people skinny again, and now they have all these tools, it might create incredible things.
In general, it's crossing into politics, but let's make a few assumptions. Let's assume that GLP, GIP agonists are safe, like really safe, which they seem to be so far. And as you said, 5 to 10 years it might take to figure out if there's any like horrible long-term side effects. But so far they seem to be pretty good. Okay, so let's assume that continues. And let's assume that the development of these molecules proceeds further, which means that the newer, more expensive versions will constantly be produced. And as that happens, let's hope that normal capitalism will kick in and the older, maybe slightly less effective or maybe less convenient versions. Like maybe you'll get a version that you can inject once a month. That would be great. But you can, for like third of the price, get the version that you can inject once a week. Exactly. So sooner or later, crossing into politics, if these medicines are truly available to people all the people who need them, not just the people who need them and can afford them, then yes, I don't think it's a question of need. It's a humanitarian question. America is overweight, and we're not the only country that is overweight. Other countries can rapidly transition into that. Generally speaking, the more wealthy the country, the more fatty they tend to be. And so if we can fix that with a weekly injection that doesn't cost a lot, which right now is not the case. Right now it still costs kind of a lot. But
well, this is something that I think we should cover is why does it need to be an injection? And where's the cost really? Is it the fact that it's a peptide, not a small molecule? Is it the fact that it's an injection, not an oral?
Medical system primarily, because Ozempic's cost in Slovakia is 100 euros.
Yeah, but why isn't it $1? Why isn't it $1?
So
primarily like aspirin is $1, 10 cents or whatever, right?
I'm going to skirt a line here because I certainly believe that pharmaceutical industry is necessary, and I also believe that pharmaceutical industry is a capitalist endeavor, right?
Well, if it's profit, in some sense, that's okay because they have to cover their R&D costs. But is there like an intrinsic cost?
Ozempic or anything else, as far as I'm concerned, when manufactured at scale, should be pretty cheap. There's no fundamental limitations. For example, Selsun Blue, right? The selenium shampoo for dandruff. If you make all the shampoo with selenium, you'll run out of selenium. So the world will have no more selenium left, and you still need it for semiconductors. So the reason why not every shampoo has selenium in it is practical. There's not enough selenium. And P&G actually faced that problem when they started developing Selsun Blue into a commercial brand. It turns out that they couldn't make enough. So they had to replace it with zinc pyrithione and so on. Anyway, I digress. The point being is that there's nothing about any of these peptides that depends on the critical thing that you can't have enough of. It's normal atoms, carbon, nitrogen, sulfur, oxygen, hydrogen, some of the most abundant elements in the universe and on Earth. Okay, so no problem, no selenium problems. Manufacturing, as we have seen, can be brought up to scale, and it's a very capitalist thing. The more you bring it up to scale, the cheaper it gets. So what is left? What's left is the desire to recoup expenses and to make profit. And so from that perspective, that's why I keep hoping that the development of these substances continues, because as you get newer and fancier, versions. They're slightly better, slightly more convenient, perhaps strawberry flavored. Then you get the older versions that are still of interest to sell, and they will start dropping in price. And as that happens, that could be very, very good for people in general. And of course, governments can choose to accelerate, governments or nonprofits can choose to accelerate that by providing at-cost medicine, like they do in many cases, right? So there are essential medicines on the U.S. pharmacopoeia that you can get for single dollars. And that's correct. That is right. That should be the case. As we see more and more beneficial effects from medicines like this, it's up to governments and big nonprofits to make things easier.
Why isn't it oral, though? That would certainly make it cheaper,
because it falls apart
right?
in your stomach.
Okay.
It's just not, it falls apart and it doesn't get absorbed. So you can make it oral by making adjuvants, but those adjuvants need to be in half a gram to a gram range.
What does
And
adjuvant
Adjuvant
mean
is, in this case, it's a substance. I'm not even calling it correctly. It's not really an adjuvant. An adjuvant is something from the vaccine world, but it's a substance that makes it possible for other things to pass through the gastrointestinal barrier. So why is it like that? It's a great question. We evolved to not die from simple stuff. So when we eat stuff, we have incredible defense systems in between us and what we've eaten. Because what we've eaten could be a bit toxic. It could be contaminated with microorganisms. It could be contaminated with soil, debris, God knows what, right? Because we've been animals not so long ago. Evolutionary speaking, we're newborns, like total babies. We've been on this earth far less than most other things of this kind. Like dinosaurs can't even compare. Millions of years we've been on this earth, not even that long, not even a whole million. The moral of the story is our gastrointestinal system is a phenomenal barrier that lets through things that are good for us and doesn't let through much of anything else. And so when we try to feed ourselves medicines, we need to permeate. So we need to get through that system. And that's not easy. So things that are the size of a peptide and the peptidic nature of Ozempic and other substances make it very difficult for these molecules to permeate.
So logically, somebody would want to make a small molecule that does the same thing, right?
And people are trying.
What are the physics of that? Let's say you want to make let's say Misha wants to start a small molecule Ozempic startup.
Right. So this is where I can actually draw something.
Let's
say that
do the drug discovery process.
Not even that. Let's talk about GLP-GIP agonists. So this is a receptor in its inactive state, and this is a receptor in its activated state. And then the peptide is bound, maybe very schematic, kind of like this.
So where's Ozempic here in the?
This is Ozempic.
The curled up.
This is part of it anyway.
The peptide part.
The peptide part, the part that matters. So it's all embedded in the membrane, and then later on there are proteins assembling on the bottom. It's what's called the heterotrimeric G protein, which then signals to the cell that interesting things have happened. Okay, so to make this into a small molecule, you have to elicit this effect. So basically a portion of this protein has to bend at an angle and lock into this conformation. So to elicit that with a small molecule, you have to replace all of these interactions, because all of this interacts with the protein with much smaller. So a small molecule on the same scale would be maybe something like this big. So it's not obvious that it's easy to do. However, I believe several companies already succeeded. So Pfizer succeeded, and several other smaller companies succeeded. It's unclear how good these molecules are. They're currently being studied. But they're small only to a sort of a certain definition of small. They're much, much larger than like a small molecule would be. Their molecular weight is, I think, in the 700s.
Okay.
Small molecule would be comfortably in the less than 500 molecular weight.
What are the physics of it? Do you think that not every part of the interaction is critical, but we don't know which one, or like how would one of
So you have to figure out what's happening here, down here, where the effect is transmitted into the next stage. And then ideally you'd find a molecule, and I think that's basically what's happening right now. You find a molecule that binds like here. Whether it causes the top to swing or not, honestly, I don't remember. I'll have to look up at the actual structures to tell you, exactly what's going on. But it is enough to signal downstream.
How do you know that it's swinging?
A structure. We've solved this. We quote unquote science has solved the structure, And we've seen
This is with cryo-electron microscopy or
X-ray crystallography, cryo-EM. Yep.
We are very lucky to have all of this history and all of these developments, and now we can treat ourselves from the many diseases compared to people who lived before us. But I hear all the time that we are about to lose part of this progress, and I'm talking about antibiotics. Could you tell us a little bit more about the situation with antibiotics right now?
It's a bittersweet story. So antibiotics were one of humankind's greatest successes, in my personal belief. It protected us from infectious disease to such an extent that 100 years ago, infectious disease was like either number 1 or number 2 cause of death for adults that have no business dying. I think it was number 1. And then of medical cause of death. And then it's number 8 or 9 at this point. So fairly insignificant. Yep. So we've gotten quite good at defending ourselves against microorganisms, so bacteria, fungi, and viruses. And a lot of the substances that have been developed during this period are very safe, on top of things. And therein lies hurdle number 1. Taking antibiotics is not entirely free of consequence. For example, when you take antibiotics because you have, a sinus infection, if you take oral antibiotics, you also might end up with diarrhea, because your gastrointestinal flora is affected and you're not feeling so great. But that typically goes away. Anyway, the point of all of this is we've gotten quite good at this. Our boundary for accepting new antibiotics have become quite high. So they have to be as safe as the old ones and as effective as the old ones. And at the same time, antibiotics are not for most people at least, they're not a chronic prescription. So you get sick with something bacterial, you receive antibiotic treatment, you get better, you go on your merry way. That's great. So from a perspective of a business, this is not as superior as someone who has to take medicine all their life. And so chronic diseases receive a considerable amount of attention because of their relevance to health and because of their economic impact. Antibiotics kind of fell by the wayside. And so there is a situation in the world where I think there is not enough new antibiotic research going on. The antibiotic research that is going on is underfunded and funded in a fractured manner. Various agencies partially fund, and you have to go after multiple grants or multiple research opportunities. And then we've had examples of commercial companies researching new antibiotics, but then not being able to afford clinical trials because they're not funded. So it's a complicated, but unfortunately primarily business-driven situation. So it's not really research-driven as much as it is business-driven. On the research end, things have gotten harder because low-hanging fruits have been picked. It's an unfortunate trifecta of antibiotics. New antibiotics need to be as good and as safe as old ones. So from regulatory perspective. From business perspective, development of new antibiotics is mostly a charitable thing and not quite a life or death or a lots of money to be made thing. Also, it's become harder because all the old ones, the good ones, are discovered already. Not to forget why is it necessary to discover new ones?
Yes.
Because it's because we are at war with microorganisms. I can talk for hours about that. And microorganisms definitely long-term are winning. So a lot of our, not everything, but a lot of our antibiotic medicines are derivatives, long descendants of natural products, which means that microorganisms have co-evolved to resist them because they are basically fighting a war between themselves. They adapt. And so when we start using them for our purposes, these genes, the resistance mechanisms, lie dormant in microbial populations until they need them, and then they rapidly spread. Thankfully, they also equally rapidly disappear, meaning that if you have a hospital situation with a lot of infections and a lot of antibiotic treatment, you may have a local flare-up of antibiotic resistance. But luckily for us, so far, it's not spreading as much further out as it can.
So what you're saying, actually, this is interesting, and I didn't know this about antibiotic resistance. It's not that the bacteria evolve randomly to resist the antibiotic. It's that they have latent existing genes that can be turned on that actually resist antibiotics because they encountered them in the past from fungi.
It's not for everything. There are antibiotics that are completely synthetic, like they never been. And for those, resistance evolves kind of like everything
randomly,
else.
Yeah.
Randomly, but with a purpose. But yes, in a bacterial world, so it's not that this particular bacteria has latent genes. No, one bacteria in a trillion may have. And furthermore, it may not even be the bacterium that you worry about. It may be some soil-dwelling thing that is completely harmless and you will never see the light of day. But it has the ability to transfer its genes horizontally. So bacteria they swing a lot. They transfer genes like you won't believe, left and right, through both the direct transfer and also phage-mediated transfer. So these viruses that parasitize on bacteria, they also have the ability, many of them have the ability to convey useful genes, useful to them, not to us. And so there are phages that, for example, can make a relatively innocuous skin-dwelling bacterium into a deadly pathogen. And that's not great. But beyond that, they also transmit antibiotic resistance genes and whole networks of them sometimes.
So basically, you give antibiotics to a person or you put it on a wound, and there's a million different bacteria species there. One of them accidentally turns out to have a latent gene that can defend against this antibiotic. That one survives, and then it horizontally passes the gene to some remaining, let's say, Staphylococcus bacteria or whatever, and then you get resistant MRSA.
Right. I would
of
fix that statement a little bit. I would say it's not likely that this is happening on a wound. It's more likely that it happens
like on surfaces or something.
More likely it happens in a waste stream. So, for example, a lot of medicine is excreted unprocessed. So if you have a sewer from a hospital and you have antibiotics in there, those can elicit a rapid game of evolution in the sewer ecosystem. There are whole rivers in manufacturing areas, especially in places that don't have strong environmental protections. So not US, thankfully. But there are whole rivers that are contaminated with runoff from antibiotic-producing factories. And so in these rivers, the ecosystem has rapidly adapted to resist stuff. It also happens, for example, when you use antibiotics indiscriminately to protect poultry. So there's a classic, I think it's a fairly classic case of Cipro being used to protect poultry. So people just put it in drinking water. Guess what? A lot of the microflora in these farms becomes resistant, and then they float around and they start spreading the good news. And again, for the most part, which is good for us, these resistance genes are not useful to bacteria outside of these stressful situations. And so they quote-unquote forget very quickly. But there's always a few that don't forget. And so when the stressful situation, like a case where somebody's being treated, presents itself, your odds of obtaining resistance increase dramatically if you have basically been in an area where resistant population is. So it's not, again, I don't think it's the bacteria don't necessarily evolve right away, and definitely not necessarily in the wound. That's too local. There's not enough of them. But if you have a patient with a dormant, slow burn bacterial infection that's being treated with 5-6 antibiotics, there's a chance that they will start generating their own resistance. And this is by no means limited to bacteria, fungi too, and viruses, but it's easy to demonstrate with bacteria. It's probably the sort of the easiest analog. Incidentally, just to keep in mind, the same exact process happens in cancer. The only difference is that in people, at least, cancer is not transmissible, which means that they are limited to one patient. It's still not great, but in Tasmanian devils, on the other hand, there are 2 transmissible forms of cancer, and they swap cancers with each other all the time. This is maybe why they might die out eventually, because there's many Tasmanian devils infected. They like to bite each other on the snout when they court each other, and these bites transmit a lethal cancer.
Wow, I didn't know about that.
Yep, 2 forms. 2, as far as I know, the only 2 transmissible cancers.
And coming back to antibiotics, it seems like we, as a humanity, we can't do anything else than try to develop new antibiotics because the process of resistant development is already can be undone.
Our coexistence with microorganisms is both beneficial and detrimental. We cannot survive without them. They can certainly survive without us. Our battle for our health will continue for as long as we are made of cells, and we're very delicious. Things would love to eat us. And so as long as we continue being delicious, there will be things out there that's going to go after our juicy flesh. And so the war continues. Luckily for us, these resistance...
What do you think of if we change all the enantiomers in our body? So instead of like everything being R, everything is L. Can we stop being edible to bacteria?
For a while, until they figure out how to do that. So as it is right now, when you eat amino acids, a good chunk of them get isomerized and then isomerized back.
Oh, okay, so it doesn't solve that.
So bacteria can certainly learn how to take an amino acid and racemize it and then eat the bits that they want. So that's chemically not difficult. So it will help, but it won't solve the problem. I think that transitioning into robots or something, that might help. And then they'll have robot bacteria eating robot bodies. It's a whole different story. So again, I emphasize that antibacterials is a story of great triumph and great sadness, because we've gotten quite comfortable with our repertoire right now, and I think we're too comfortable. And the awakening might be the next pandemic, except it won't be a virus like COVID. It may be something bacterial or fungal, and it will not be pretty. Again, we probably will catch up, and eventually, a couple of years, there'll be new treatments, but a lot of people will suffer in the meantime.
I was looking for some information on the topic, and then according to in the last 5 years, there were only 12 antibiotic drugs released. But then my question would be, so it seems like the frontier of drug discovery is moving maybe towards other areas. Could we look into the direction, like what is now the most hype topics in drug discovery and where most of the companies are trying to focus on?
Inflammation and immunity is a blue ocean, is a frontier of great discoveries to be made. And a lot of it has to do with us living longer and starting to be interested in taking care of things that previously may have been relevant but not life-threatening, or life-threatening but at a longer term where people just didn't have the time to get to them. So now we have time to get to them and the knowledge. So that's certainly a very exciting time. It's been going on for 10 or 12 years already, maybe 15, and it will continue for a while. The very, very hot and perennially hot topic of gene therapy, how to make these changes to our bodies, how to make them such that we get what we want and not what we will occasionally get randomly. And of course, the study of longevity itself, how to have long, healthy lives, or how to have healthy lives longer, even if we overall live the same.
What's the reason for aging? Why does aging exist?
Because there is no point, evolutionary speaking, for us living forever. We exist to make kids, and then kids make more kids, and by age 40, we're done. So from the perspective of us being little animals, there's really no benefit. Now, of course, again, this is just evolution of living things. So that's not necessarily taking into account the fact that we can think, and so therefore our evolution has a lot to do with mind and not just body. But from the nature's perspective, our lifespan as it is kind of long, right? So
do you think there's something fundamental about the limit, or is it arbitrary?
It's not arbitrary, but it is difficult. So the case in point are the classic dependence of lifespan on size. So generally speaking, larger things live longer, like whales and such, like giant turtles and so on. And yet there are bats that live 40 plus years, which for a bat is insanely long. So why and how? People are very interested in that right now. People are trying to find out, and this is where the two topics intersect. There's a considerable degree of interest in these bats from the perspective of their immune systems and how the immune systems are dialed and functional. And that is possibly related to why they live these unusually long lifespans. However, at least in theory, there's really no reason why living species have to die. And it's mostly, I think, again, evolutionary benefit, just not living for too long, leaving space for the progeny, having progeny live a fixed amount of time, leaving space for the next one, so the game can continue. Otherwise, things that live too long maybe have died out from changes in environment that were rapid enough on their life scale that they couldn't adapt. So animals adapt by evolving. Individuals perish, the species persists. We are still just animals.
So if you want to make a drug that, let's say, doubles human lifespan, it's very difficult to do studies, right? Because they have to be very long if you want to target the lifespan. So what's the proxy? Like, how do you, let's say today you have $1 billion. How would you double the human lifespan with $1 billion?
So I'm certainly not qualified. I'm just a humble research scientist, but you have to start. So people have started with models. So people figured out how to make mice live quite a bit longer, how to make nematodes live quite a bit longer.
Quite a bit is...
Quite a bit is like long, like 30% longer, for example. Some of these tricks involve restricted calorie intake. You can live a fairly miserable life and eat just barely enough to survive. And if you're a mouse, at least, you will live longer. But it's difficult to imagine for people that being a very successful therapy because that really stinks. You have to be really hungry like all the time.
Maybe Ozempic will help us.
Maybe.
But I've heard at least that these things don't translate well from mice because mice are usually predated. So they're usually eaten by another animal. So they don't have a lot of longevity kind of mechanisms because living longer in theory doesn't really help them because they get eaten.
Well, the control animals and the test cohorts are all living nice, comfortable lives in isolated cages. Nothing's eating them.
Yeah, but my point is that the biochemistry of mice might be poorly adapted to living a long time because they're predated frequently. So there's no like evolutionary, you can't keep having kids because you just get eaten. And so I've heard that it's easy to extend their lifespan relatively because they don't have a lot of mechanisms that are low-hanging fruit that might already exist in longer living mammals.
I think that...
like maybe they get cancer more easily, they get this more easily, that.
Well, one would have to look at what mice die from in old age. What people die from in old age, there's the classic three: cardiovascular, CNS, and frailty, so to speak. So multiple systems failing. How is that to be fixed or prevented is a very, very open question. I'm sure you know that more than I do. Billions of dollars were put into several startups, and that's just the ones we know of.
But what do you think is like the most
out
promising, if you had to guess, like if somebody solves this problem or like really makes progress on it, what area do you think they'd be working in?
Personally, I think that this is purely personal thought, is that addressing the zombie cell phenomenon, which you may have heard of. It's the cells that have extended their kind of life beyond their natural state, and they sit there. They don't die, they don't divide, but they do secrete all kinds of messengers that locally and globally affect the overall system. So addressing that problem is a very good start. But again, I'm not qualified to really tell you where exactly.
I don't know if anyone's qualified. Nobody has the answer, right? So it's
Well,
kinda...
Otherwise, it would be solved already.
So
Well, also like for longevity, I guess like the main question is why would you want to live longer? Because on one side, as I understand, like from neuroscience perspective, your brain always degrades. So why would you want to live longer as a vegetable? So you want to
No, the assumption is
if there is a way to replicate neurons and get new ones.
the assumption is that all of these things somehow would preserve your brain also. Like if you solve this problem somehow you would. I mean, if you look at it like it's your brain lasts, let's say 80 years in a reasonable way. Why 80? Like it's very arbitrary. Right? It could be 160.
Yeah, but you see
or something.
the quality of life I think drops quite significantly after. So I guess now like people started to live longer, like even my grandparents. But I wouldn't say that extra decades of life they live, it doesn't really contribute too much to because they wouldn't participate that much.
The longevity
in society.
people, the like kind of longevity optimists, their argument would be that it's just an arbitrary number that you're tuned. It's like you're tuned to die in this period of time, maybe evolutionarily or whatever, but the number's arbitrary. If you could find the knob, you could tune it to 50% higher and everything would just stretch.
Okay.
So let's say maybe your adolescence would stretch. I don't know. Your adolescence might stretch and then your old age might stretch.
From my perspective, I would prefer to die right after I'm like not really contributing to the society. I'm kind of useless, and then why would I want to leave and be like
We'll ask you later how you feel about it.
But anyway, with the prolonged life, the age when we are active also prolonged. If you can see older people right now lead perfect life, amazing life.
that's why I brought up the staying healthier longer. Even if the lifespan is roughly the same, just being able to function at your 70% capacity rather than a 30% capacity, however you define percent, when you hit 70 or 80, that would be already great, given how difficult it is to be old.
The physics or whatever first principles argument that I like, that I think probably other people talk about, but it's if you take like a 40-year-old woman and she has a child, right? The child still comes, and the father is also, let's say, 40 or whatever, so the child still comes out 0 years old. And it's actually made of the parents. It's not that the parents make it in a vat, right? It's actually made from the cells of the parents. So there's definitely some kind of potential, and that's been going on every generation. So
Although admittedly,
actually we are
the
like,
older the mother, the worse the odds for certain things are.
yeah, for sure. But in principle, like we are, like my cells are actually just a continuation of some billion years ago cells, right? Or even if you just take humans, the first human, their cells are in me in the end. So that means that in a sense, it's not that at some point there's a reset that's like a hard reset, like the cells are made again from chemicals. You take vats of glycine and pyridine and whatever, and you just mix them up and you make a human. It's always from a cell. So
Well,
there is,
however, a bunch of reset events that happen during fertilization. So the epigenome in particular gets basically reset, which is hybrid vigor, right? An interesting thing, you breed two plants of related but distinct lineages, and you get the first generation cross, which is amazing, like so lush and just amazing. And then it splits into subcrosses later on, and they're not so amazing anymore. A lot of that has to do with epigenetics. So the reset of epigenetic being information that is recorded on DNA, but not in the genetic code. So DNA gets acetylated, methylated, and all kinds of other things. Plus histones themselves get acetylated and modified in various chemical ways. And that is information that is heritable, but during embryogenesis, a lot of it gets reset. So we are not just sequence, right? We are also epigenetics. And that's the hard one, really hard one to address. Not to mention that we are also all of the microflora that we are, right?
So you mentioned plants.
Mm-hmm.
I guess maybe they come to mind for you because of your long time at Monsanto. What's it like to work at Monsanto? What was it like? Some people consider Monsanto the root of all evil. What's it like to work at Monsanto? What's your perspective from having worked there, and any stories that can add some color, because it's kind of a black box for most people what
Right.
happens
So
at these large companies.
Monsanto is no different from pretty much was, because it doesn't exist anymore. It was bought by Bayer and assimilated. Monsanto was no different from any other large pharma-like company that I've worked in. It was driven by the same basic business principles. It was not more ethical or unethical that I could tell. It was pretty average. And we certainly, at least on the level that I was exposed to, we did not set out to do harm. We set out to increase productivity of plants and to make farmers happier by giving them more yield and better resistance to disease and so on, but also to make our shareholders happier, right, by selling more seeds and integrated farming systems. So I think that in the end, the Monsanto had experienced several unfortunate things in its history that predisposed the public to recognize its name. And those things were, for example, the whole Agent Orange story, where it was at the time where Monsanto was still doing chemistry. Essentially, the long story short, the government insisted on making Agent Orange in large quantities and ahead of schedule. And the government...
What was Agent Orange?
Agent Orange is a defoliant. So during the war in Vietnam, people were hiding under canopies, and defoliants were used to expose them so that we could bomb them.
So the leaves would fall off the trees.
The leaves would rapidly fall off the trees. So Agent Orange is a fantastic defoliant, but if you make it...
How does it work, actually? It's like a signaling molecule that tells the tree to drop the leaves, or what's the...
It's part of it. Part of it has to do with light damage. It's not a simple process. It was discovered by accident, for certain. Anyway, the more interesting part is that when you make it as per spec, the amount of toxic substances in it is low. When you accelerate production, it becomes high. So I don't have physical evidence of that, but anecdotally, the supervision by the government at the time caused unreasonable acceleration. So process temperatures were raised and so on. And so things became laden with the terrible stuff that caused a lot of people to be deformed, and all kinds of problems arose. So Agent Orange was one part of a problem. Then the unfortunate choice of a name, they called something Terminator, which was basically a system designed such that seed can be planted for one generation, but you cannot harvest the seed and replant it. And they called that Terminator. That was an unfortunate choice of a name. And certainly it displayed a degree of greed, right? So the idea behind it is you should really not steal patented seed. You should buy it from us, whoever us is. Now, the interesting thing is that most large agricultural companies have stuff like that. not terminator. I mean, they most have desires to be the sole provider of seeds to a particular farming community or a particular farming line. And it's going to continue as long as capitalism is the dominant force of economy, just how it is. So if you are beholden to the rules of capitalism, you're going to have stuff like that. When you find something better, like economic system that's better, please call me. I would love to be there. But I've seen the alternative. It does not strike me as good. So socialist economy is not a good way to do it for large scale. And I just don't have a better alternative at the moment. I don't love it, but it's how it is. I don't find capitalism to be particularly friendly towards sick people, old people with young children. There are people who are affected by life unfairly, and capitalism doesn't really work for those guys.
And do you think that the capitalism may have already led or will lead to some point when there will be like several companies competing with each other? One will produce synthesize new types of diseases, and the other ones will try to block them or find a new drug. So it will be a competition between several entities of...
This is a little bit science fiction-y, so I'll try to answer it in the same jovial mode. In order to make a disease, you have to understand how a disease works. Thankfully, for the most part, we are still quite far as civilization from the ability to truly make designer diseases that will do our bidding. I'll tell you what I'm worried about. I'm worried about the kind of government like, let's say, North Korea, where there's no control, no ethics, no will of the people, so to speak, only a will of a few, and a desire for total domination of, let's say, their own population. So some unscrupulous person may appear and say, oh, come on, I will give you this really nice virus that will make you guys have a population that is docile and compliant. And this is great, they will say. You won't have to put people in prison anymore. You won't have to beat them with rubber hoses in dark rooms. You can just spray them gently with this virus, and they will listen to every word that you say, and everything will be great. And the odds are that it's not something that can actually be made. So they will make a mistake.
Sounds like some kind of totalitarian Ozempic.
Right. I warned you. I warned you.
It's probably would
I
have
warned
to be
you.
a
We're going to
peptide.
go science fiction here, deep science fiction.
I mean,
stuff,
Ozempic
okay?
would have been science fiction 50 years ago. If I wrote a book 50 years ago that said that every American would be getting an injection every 2 weeks to control their appetite, like in some kind of magic way, right, to turn them into a monk who doesn't like to eat, that would be science fiction.
And yet, even 50 years ago, look up fen-phen, the combination of two drugs that causes you to lose weight rapidly. Unfortunately, gives you horrible heart problems. If not for the heart problems, we'd already be all thin.
Is this the drug that screws up the mitochondrial membrane?
Among other things. But fen-phen it's not quite the same. But you're talking about nitrophenol. If you remember, there were factories where people would paint. So there was a radium story, but there was a particular one where people were using this yellow paint, which was a dinitrophenol base.
And they all lost a lot of weight.
They licked their brushes. They lost a lot of weight. And also they had like horrible problems. So even 50 years ago, there was this strong belief in the power of science. So I don't think that they would have been that surprised. I think they would have been, however, surprised that we would get so fat. Right? So 50 years ago, they weren't so fat. So it may have been surprising to them to say, what? Every third American or every fifth American is morbidly obese and every second one is pretty fatty. They would be surprised, I think. They'll be like, what's wrong with them? So again, imagine a country where you have a dictatorship or a totalitarian regime of some sort, and they really want to control their people, from the inside. What better to say than, oh yeah, let's make this virus or something that will make them non-confrontational, that will make them totally obedient, and they'll make them, super attentive to the message of the party. And then, God forbid, somebody actually does, instead of just taking the money and running, which would be the preferred option, they would actually go out and start genetically engineering some kind of a viral vector. Without poor and incomplete understanding of the human brain, what does it mean a person is being more obedient? Which gene do I turn on? Nobody knows. There's no gene for obedience. It's no nonsense. There's whole networks of things that create a personality, upbringing. It's all a mixture. But nevertheless, if somebody tries, what they will create more likely than not would be a deadly pathogen. And then it'll get out of control, decimate the intended audience, and then spread around, and we'll have a COVID on steroids, if you will, right? So that's what worries me. It's places which don't have enough control and don't have checks and balances in place, like the actual scientific community is for the most part quite good at controlling itself. We haven't created super pathogens. We haven't created like two-headed monsters. This is good. This is good. So for the most part, and especially in a country where you have a lot to lose, right? So living in a nice chubby American culture. But if you have not much to lose and you have ambitions of grandeur and very low ethics, I can see a situation like that. Much more likely than some evil pharma company. It doesn't make a lot of sense for a pharma company to create a new disease. It's cute, it makes nice stories, but it's much easier to make a nice drug because it's really hard to make a new virus to begin with. So much easier, much more profitable. Plus, why would you want to kill your consumers? That just makes no sense, right? So it's that kind of self-check of the capitalism that gives me a degree of comfort, but only a degree, right? So humankind is much more curious than that. And so will we eventually come to a point where we start making things that put us all in jeopardy? I think the same has been said 100 years ago, right? The invention of nuclear weapons, the invention of the internet, the invention of AI, the invention of new forms of life eventually, that all of that is coming. We have to deal with it. But our goal is to create a society with enough common sense that when we have to deal with it, we deal with it right.
I have a question for you that I think it's a kind of a 2-3-part question, which I think is interesting. First of all, for people that are not really familiar with drug development, but are familiar with AI and machine learning and so on, kind of like people from the tech world, there's a lot of optimism about using AI for drug development. And there's also a second kind of layer on top of that. Once you get optimistic about that, which is an assumption, there's a second layer, which is, wait a minute, if AI gets powerful enough, it can start making its own drugs, pandemics, whatever. So I think for me, as somebody who's like worked in a wet lab, I'm always, this sounds real. I'm very skeptical of this, but I don't work in drug discovery. So I think it's an interesting question for you is, first of all, what does it take to, it's kind of a 2-part thing. When you're developing drugs, what would it take for AI to actually be useful for that? And the second thing is, let's say you want to take humans out of the equation. Let's say we want to make an AI Artem, right? So you set up this lab that we're interviewing in that's next door to this conference room. What does it take to set up the lab? Let's say I'm an AI. I want to build a lab and make a drug, let's say for good or evil. What does that actually take? And what would AI have to reach, or like what kind of data would it have to have access to actually develop a drug?
It's a very complicated question on multiple levels. So let's try to dissect a little bit. Let's start with information. Previously in today's conversation, we've touched on the topic of incomplete understanding or very poor understanding of life as it is. And we do have that problem. So we can only teach the AI what we know ourselves. So by definition, AI would have to have access to the totality of human knowledge pertaining to physics, math, biology, chemistry, and so on, right? Let's assume that that can be accomplished, which so far it hasn't been as far as I know. So far, AI has been trained on internet, which means cat cartoons and like best recipes for rhubarb pie, and it's great. I hope it stays that way because I am dubious about AI as a phenomenon, meaning we can be digging our own graves here a little bit, but we'll see. Anyway, so you give AI access to all of that, and then you have to give AI effectors, right? So you have to make AI able to physically perform the types of operations that are performed in a normal drug discovery, molecular biology, biochemistry, chemistry lab. So you have to equip it with effectors capable of synthesizing new chemical entities, testing them on model systems, and then repeating that in a sensible manner while augmenting its own capabilities as it goes. I think it is entirely possible, but it is very difficult. So right now, the effectors that we use are all targeted towards humans. So just to give you some small example, in order to move a few microliters of liquid from one place to another, we use a pipetter, which is a handheld device with a disposable plastic tip that comes on. You suck a little bit of liquid into it, you dispense it into a new place, and you throw the tip away. That's kind of become the standard. Is it the best thing for AI to do? I suspect not. I suspect that if you don't have hands or arms to begin with, and if your eyes are not at the level of roughly 1.5 to 2 meters from the floor, and if you're not limited to any of this, then why the hell would you need a pipetter?
Maybe lab on a chip?
Sure, lab on a chip, whatever. My point is that it's just the basic operations of transmitting a portion of fluid from one location to another, viewed from a perspective of an entity that is not restricted to human anatomy, is a task that ends up in places where we probably don't know. So I cannot tell you that I know how it will look like. I can tell you it will not be a pipetter. But to get to that point, the AI has to first go through iterations of doing it first like people do, right? So we can only teach it what we can do. And so what I am very curious is AI fundamentally going to be able to develop that type of a progress? So is emergence a real thing in AI? I think it's a hotly contested subject right now. The concept of emergence in AI is not at all clear, but it would have to be, because given a pipetter, generally speaking, a machine would do a crappier job because it's not a human. It makes no sense for a machine to develop an arm with which to hold a pipetter, push a button. That just doesn't sound right. I would be very interested in seeing what would ensue, but I guarantee you it will not look like something that a person can use.
So even if you step back from the mechanical part of it, you have to think of what to do, right? So there's I'm sure a lot of AI drug discovery, I don't know how long people have been pitching AI drug discovery, at least 10 years, right?
Yeah.
And before that, maybe it was called computational drug discovery, right? But it's really in some ways the same thing. There are no AI discovered drugs.
Not that I know of.
Or maybe not even, I don't know if there are even like good candidates. Maybe
there are companies that proclaim to a greater or lesser degree, including our own, that AI is helpful. So we think AI is helpful, but you cannot dispense of humans.
But it's helpful
so
in the
far.
sense of coming up with some suggestions for molecules, or like where is there any traction at all?
I think we've seen traction on molecular design. So yes, suggestions of new molecules based on previous molecules. I think there's a good deal of traction in design of clinical trials and interpretation of clinical data, and the ability of machines to really not get bored with a deluge of small details. So I think that we've seen a lot of interesting things happening where machines are looking at complex, multidimensional biological data. So a combination of biomarkers and biophysics and biochemistry. You can produce an absolute deluge of data from a few cells, if you want. A single-cell sequencing, single-cell proteomics, you name it. And the point in all of this is that a human, which is what we touched on before, is limited by our ability to absorb and interpret data and hold it in working memory at one point, whereas a computer doesn't seem to be limited at that. I think the computer is currently limited to the whole emergence thing, the imagination, the creativity. And if a computer can overcome that boundary, I think we will see sort of true AI developments. Because as you said, abstracting from mechanics, you can say, okay, AI is going to work like this. It's just going to have a dedicated team of people doing all the same thing that people do, but completely brainlessly, right? Like, go make this molecule, human slave, and they'll just go make this molecule, and then go test it on a rat and tell me, did it squeak? Did it twitch? What happened? That obfuscates the need for emergence from that perspective, but the need for emergence remains.
But isn't there, it's actually an interesting question, because there's a trend, like if you look at big pharma, they do everything in-house, more or less. Maybe that's not even the case anymore, but if you look at startups like Origin, actually you do use a lot of CROs, right? So in a way, that's already a trend towards centralizing the intelligence component, like the expertise, and outsourcing the manual work.
Yeah, I'm still dubious as to how well outsourcing is serving us. It's an economically made decision for the foremost. So I am not sure that outsourcing is to the best interest of pharma, personally.
When you say, you mean like outsourcing the production of compounds, or?
So we do it because we have to. There is no way that a small company can absorb and integrate every aspect of things necessary to develop. I'm not even going to talk about a drug. I'm just going to say drug candidate.
Mm-hmm.
Just because if you want to test something in mice, well, you have to have a mouse house. Are you going to spend money building a mouse house? Chances are no, because you can rent that expertise from somewhere else. Sounds great. However, will that exterior expertise supply you with the same diversity of data and the same amount of data that you didn't anticipate?
Mm-hmm.
So if you're doing a study on mice and you just want to know if something is toxic or not, you will get that same data, right? They will say, okay, so you've had a 25 cohort mouse, you gave them each 200 milligrams of your stuff, and 24 survived, 1 died. We did an autopsy, its liver was large and its heart was shrunk and it was pretty sad. That's fine. But what they might not tell you, because you didn't ask them, whether the other 24 mice displayed interesting behaviors. Did they run in circles? Did they sweat? I don't even know if mice sweat. I don't think they do. But the point being is, was there something else?
Because there's a loss of the serendipity of science.
There is absolutely a loss of that. A CRO is not encouraged to do that. They're paid to do a certain thing. The best they can do is to deliver that thing and then not make too many waves. That is where integral abilities may be better.
About the startup where you work, Origin. So what's the structure of it? What's the main goal of it, and how do you integrate in the whole system of the drug discovery?
So we're small. We're 12 people, and we want to find new drugs for inflammation and immunity. And we want to do it using a combination of ultra-high throughput screening. So DNA-encoded libraries is a form of very, very high throughput screening. I can explain later if you want what it does, but basically exposing molecules, billions of molecules to your target and then picking the ones that stick. That's part of it. And then our big other part is using a form of machine learning to derive kind of nonlinear benefits from the output. So develop better molecules faster. But because we're small, we, at this point at least, we're not dreaming of going past a clinical candidate. We would definitely want to have a partner for that. And
that's the typical infrastructure.
It's a very typical case, but what makes us different is that we are not really bound by targets. When I say inflammation and immunity, that's what we're currently working on. But next year we can switch and start working in CNS, or anything that we can isolate a target for and have it physically in our hand is basically subject to this method.
And the difficulty in the high throughput screening methods like DEL, in some ways, it's not even the screening, it's really having the target. It's being able to have a stable version of the target to drug.
For some targets, it's a relatively solved problem, I would say. Relatively solved problem. We're pretty good at this point, including we being here at Origin. We're pretty good at making our targets. The main difficulty, so there's multiple levels of difficulty, but the main difficulty always is to find high-quality initial hits. Because even if we have billions of molecules, there's no guarantee that our molecules will contain the molecule we want.
How many molecules exist in like the Platonic world? Like how many is it? 10 to the what?
Oh, total? It's immeasurable. Numbers much larger than number of atoms in the universe. It's exceeding 10 to the 60 easily. And that's only molecules that sort of follow sensible chemical rules and are made of the first 5, so CNOSH. Just these molecules, when you reach like the count of 12 or 15, you're out. Like there's not enough atoms in the universe to make them, just one copy of each.
So would it help to use, like for example, quantum computing somehow to get to those high numbers?
Once somebody can explain to me exactly what quantum computing is, then I will be able to answer this question. Right now I plead ignorance. I understand some basic principles of it, but I can't quite see it working well enough. When it cracks the cryptographic code, then I would be like, okay. But it hasn't done that yet, so I'll just wait. It's not about the scale. It's about being able to replicate inside a computer, quantum or otherwise, with good enough accuracy what's happening in real world. Because if you think about it, a test tube is a quantum computer, right? So I have a gram of substance in a little tube, and I add some stuff to it. It performs essentially a quantum computation by binding. So a piece of something binds to a piece of something else, and then I read it out by spectroscopic means or some spec or something, right? That's a quantum computer right there. It's just made of the same atoms as the rest of us. To me, it's not that difficult, and actually costs a bit less, right? So you don't need to cool anything to like crazy temperatures. The trick is...
Yeah, the real quantum computing is happening here at Origin,
I think it's happening inside us every living second.
Yeah.
So we are quantum computers.
Well, maybe it's like with quantum computer, you can get inside to see what's the
I
level structure, what is the processes.
I think the difficulty, if you talk about it practically, the difficulty is that when usually you care more about the interactions of let's say a small molecule and a protein. And so for quantum computer, modeling a small molecule would be like a big deal. But if you also have to model the protein, that feels difficult because let's see, like a protein is how many atoms, right? So it's
10,000, let's say.
Yeah, let's say so protein's 10,000 atoms. But it might be that you can abstract that away somehow and just look at little sections of the protein at a time. I think people have struggled with this for a long time. So how many qubits would it take to model 10,000 atoms? That's I would say like the first
10,000. Is
it literally a one-to-one? I don't know about that.
I think it depends how do you formulate the problem, and maybe there is a way that you...
Because it's not like a crystal or something where each atom has like a single, it can be, let's say, spin up, spin down or something. You have to model their the force fields or something between the atoms is probably what's important in chemistry.
And the whole idea of a force field is an abstraction. So the problem that we have, regardless of which computer it is, quantum or normal, is that the best option for modeling molecules seems to be quantum mechanics of some sort. Because of computational expense, typically people go with density functional theory and its relatives, because it greatly reduces the computational load. But even on that level, modeling whole proteins interacting with small molecules or with other proteins over a period of time is expensive. So what people do is they do an even further approximation. So we're talking molecular dynamics. So in molecular dynamics, atoms are balls. There's such a thing as bonds, which is, of course, as we know, bonds are abstraction. There's no bonds. It's just electrons being smeared across things, right? But bonds are convenient abstraction. We like to draw them single, double, triple, right? And these bonds are approximated by springs. Then there's, for some reason, atoms follow laws where angles are fixed. And then dihedrals are less fixed, but still fixed, so angles between four atoms. And then all of it is somehow held together. And then when atoms are not bonded but are in contact, then you have the quasi-van der Waals law, right? So you're not supposed to interpenetrate. And then there's some attractive interactions and some repulsive interactions. All of this is a soup of what we consider chemistry, but it's on a very abstract level. So it's nowhere near what actually happens. And not surprisingly, unless you're exceptionally good at it and you're exceptionally patient and you dial parameters for a single system for years, most of it is underwhelming.
It's not predictive. Basically.
It's underwhelming. You want more out of it and you get very little. So I am a big fan of quantum mechanics computations, and for small molecules, they are absolutely the best you can do, especially when you do real, serious, hardcore quantum mechanics with no compromises. But even that tends to model a single molecule of compound in vacuum at best, or in some very, very simulated solvent, like harmonic restraints with boundary conditions type of solvent.
Or at zero temperature.
Well, no, you can model at real temperature with some trouble.
Yeah, well, like of course it becomes more and more expensive.
Yeah, it's super expensive. And then the heavier the atom gets, it's more expensive because you have to model more orbitals. It's hard. I think it's doable.
Who's doing all this computation for us to exist? Like, how is this?
Well, that's part of the beautiful part of it is that it also sort of seems to happen by itself.
Yeah, it's very suspicious.
Well, again, that's why I made a facetious analogy between quantum computer and a test tube with stuff in it. But it's not all that facetious. It may be cheaper to do the thing than to simulate a thing, you know? It may be actually cheaper to just do it than simulate it, if you need the degree of simulation that will give you the tangible, actual results.
Yeah, I guess the difficulty with doing the thing is that the readout method, so like in the case of a simulation, your readout method maybe doesn't disturb the real outcome, but in the case of the experiment, your readout might disturb the chemistry of the system, right?
That's less of a problem. The bigger problem is that you can simulate single molecules interacting with single molecules and get away with it. In a test tube, it's pretty hard. So when you start dealing with single molecules, then suddenly simulation is cheaper.
Yeah.
But of course, we use ensembles, right? We use billions of molecules. Like our smallest volume that we do experiments with is, say, a volume of a cell.
Well, but I mean, like, for example, if you take DNA-encoded libraries, and I think it's good if you can explain it a little bit just in general terms, but every one of those billion molecules that you're screening, that's attached to a DNA barcode, right? So it's not bare, it's not like on its own, right? And so in that sense, the measurement system or the screening system itself is affecting the outcome.
Well, of course. So for the brief introduction into DNA-encoded libraries, the way it works is like this. You start with a short piece of DNA, and I'm going to draw it like this. And this is double-stranded part, and this is kind of single-stranded-ish. Here is a modified nucleotide on which is a linker. So this linker is of certain length, depending on the method. You can have longer or shorter. And the linker is typically made of a substance called polyethylene glycol, right? So it's got this going on, right? And this linker is generally water-soluble. Polyethylene glycol is, by the way, what you take when you need to go, number 2, and you can't. You take it, goes through you, accumulates water in the colon, and you go number 2. Anyway, so water-soluble. On the end of it, there is a synthetically friendly chemically reactive group. So I drew an amine, for example. Okay. First thing you do is you take this amine, and I'm going to now greatly simplify. So I'm going to still draw the same thing, but I'm just going to say NH2. And now I'm going to make an amide out of it, right? So NH2O, right? Then R, and then N, and I'm going to put a star here. This star, it represents a protective group. So this nitrogen is not reactive. Otherwise, it'll make a polymer. So having done this, I'm going to say my R, my amino, this is now an amino acid, right? This part is an amino acid. This is the amine. This is what used to be the acid. There's many of these R's. Let's say you have 100 of these. So R1 through R100. You've done these reactions in isolated little tubies or in wells on a multi-well plate. And each time you do a reaction with a particular amino acid, you also ligate. The word is ligate. It means attach in DNA world, chemically attach or enzymatically attach, doesn't matter. Another piece of DNA. And this piece of DNA is what is the first part of the barcode. It encodes this number. So whatever, RI, right? So this is I. And again, you do it 100 times. So then you take a whole bunch of this and you mix. So now you have all of this together mixed. Then you deprotect the N. So the next step is you take this mixture, you split it again and say again another 100. And now you have first barcode, then the thing, which we call the headpiece, N, right? I'm going to again simplify R1, another N, and now it's available. So now we're going to put R2. And R2 is also 100. So again, 1 to 100. And we attach a second barcode here. So this is I, this would be J, right? So this is barcode J. And again, we mix. And then we do it a third time. Maybe even fourth, depends on how creative.
So each time you're getting a permutation of the different R's, and every R you add the barcode for that R to the DNA.
Right. And so at your end point, I'm simplifying a lot, but let's say you have I, J, and K. And then you have your piece of DNA, which has essentially 3 building blocks. And again, your I, J, and K. One is like that, one is like this, and one is like that. And all of this is mixed in one tube. So if you do it 3 times, you get 100 times 100 times 100 actual different molecular species. I'm greatly simplifying, bear with me. So then you take this entire mixture and you expose it to your protein, which I'm going to draw like a Pac-Man.
In this world, you have a million. This is a million. In the 3, if you do 3 steps, you have a million.
100 times 100 is 10,000, and another 100 is a million.
Okay, so you would have a million different molecules
molecular
with
species.
Just these puzzle pieces mixing up in a million ways.
So there's not a million molecules, it's a million species. The amount of each molecule is going to be more than one.
Yes.
Because if it's just one, you're kind of screwed because you're going to lose half of them.
Each species has its own barcode, basically, so you can later pull it out.
So you take a piece of resin, a solid support of some sort, soluble, not soluble, but suspendable in liquid, and you attach your protein to it with some kind of a linker. We can use a tag or a chemical linker, doesn't matter. So a protein is a Pac-Man, and it wants to bind this particular molecule, which happens to be, I don't know, I-137, J5.
And so in the practical world, this protein is, let's say, some inflammation receptor, and you want to block that receptor, so you're searching for some small molecule that will block it from this ensemble of a million.
Yep. And so we begin by basically exposing this protein to the entire million, and then somehow physically separating things that bound to things that do not bind. And so the things that bound get retained on the protein.
So you wash away everything else.
You wash away all the other stuff. And here, of course, friendly physicists will remember that you're dealing with association and dissociation thermodynamics. So if you take long, then it will equilibrate off. So tighter binders will take longer to equilibrate off, but they will eventually come off. So you have to do it kind of fast. Once you remove the mix, you have to do it fast. Otherwise, your equilibrium will go, it'll go into solvent, basically. So at that point, you basically dissociate them with heat or with a denaturing agent or whatever you want. And you then PCR amplify the barcodes. So each barcode ends with a place.
This is an important thing that I think will be missed by everybody that's not familiar with this. So it's so PCR is this magic Nobel Prize winning technique for amplifying DNA. So you have basically it works almost with one unit of DNA. So you literally have one molecule of DNA, and then you can replicate it correctly, like with very high fidelity, into basically infinite quantities. And how this is done, it's done using the nanotechnology that replicates DNA in bacteria, plus some very clever human hacks. It's kind of like a airplane in a sense, where it's not exactly what happens in biology, but it's inspired by biology, except it's like an airplane that uses real feathers. So you pluck a bunch of feathers from animals and you turn them into wings or something. So it's using the actual enzymes from bacteria, but with very clever like temperature cycling to create this, to create the copying effect.
But is actually PCR still state of the art, or?
Very much, yeah.
It's as practical as this pen. If I'm a biologist, if I don't have PCR, it's like I don't have a pen. Like, sorry. PCR is just fundamental to our work. There are other things that you can do, but they're not as good yet.
So overall, let's say when you would like to find a new drug, how many experiments do you need to do in order to find one?
Wait, before we cover that, just to let Artem finish, they use PCR to amplify the DNA barcode.
Yep. Then we sequence the result, and we sequence in what would be called the next-gen sequencing unit. So some kind of a machine, typically made by Illumina, which can produce, let's say, 1 billion reads. So you put in so many molecules of input, and you get 1 billion messages back saying you had this molecule, and you had this molecule, and you had this molecule, and you had this molecule,
all DNA
so on,
strands.
so on, so on.
It's reading
right?
the DNA.
All DNA. So then you decode it, meaning you apply the same rules that you got built into the IJK, you apply them backwards. And from IJK, you get building block 1, building block 2, building block 3. And that is a recipe for a molecule. Now, it is very important, critically important to note here that it is not the molecule itself. It is an information of how a molecule could be built. And why is it important? Because when you do chemistry on DNA, you sometimes get surprises. So chemistry doesn't want to go the way you think it will. It might go some other way. And of course, all of these reactions are highly optimized to give you what you want, but it's not always the case. So you have to keep that in mind. So that in the end, you get a list of molecules that you then have to make and then expose to your protein and find out which ones of them, if any, interact with the protein in a meaningful way. So that's a DEL in a nutshell. There's many technical details that I've omitted. The fact that we do more than one round of selection, for example. The fact that these libraries are not one library, but mixtures up to 100 or more. It's relatively inconsequential that I don't say it. But if you're going to practice DEL, all of these things, of course, come up. And in the end, you get a bunch of recipes. And this is the critical part where something like AI or ML could make a big difference, is that if you don't have to make them all, that would be great, because making a molecule is expensive. So one molecule costs about $2,500 to make, and you get maybe 15, 20 milligrams of it, and it takes 2.5 months. So if you invent your best molecule today, you draw it on a piece of paper, and if it can be made, because not everything that can be drawn on a piece of paper can be made. In this case, it's not a big deal because you've actually made them already, because you made them on DNA in the first place. So if you write on a piece of paper, I want this molecule, and then you send it off to China or Canada or US, wherever you find your friendly chemist, then they will take, on average, about 2 to 3 months to make it. And then they'll send it back to you with a bill, and they will be like, here's your molecule, give me $3,000. You give them $3,000 because you want them to do it again, right? You don't want to cheat them. And then you go test that molecule. So from ideation to testing is at least 3 months. That sucks, right? That's this, and this is a basic unit of drug discovery, no matter what you do. If you don't do DEL, it's fine. No matter what you do, from ideation to knowledge is about a couple to 3 months. Larger companies have developed interesting and fun ways to make things faster. We are working at Origin on our own interesting and clever ways to make things faster, how to make molecules faster, how to make smaller quantities of molecules faster. That's all very, very relevant to our life. But in the end, this basic unit of invent something, make it, and test it, it's about 3 months. That's why it takes so long. Let's say you invented a molecule, you tested it, and it's like, it's pretty good, could be a little more potent, Also, I noticed that it has the solubility profile of, a Sony Walkman. Like, it's a problem, right? If something doesn't dissolve in water and your enzymes are in water, that's not good. So I would like to make some modifications, you say. So you go to your friendly chemist, or if you're a chemist yourself, or aspirations of chemist, you design an improved molecule. Let's say you add a carboxylic acid group to make it more soluble. It's another 3 months. And then it comes back, and then you find out that, oh, crap, it doesn't work. So I added a carboxylic acid. I thought I was ever so clever. No, it didn't work. You ruined it. You made it bad. So, of course, nobody does it like one at a time, right? You design a cloud of molecules, and this is where money comes in. So depending on the size of your money, you'll get a bigger or a smaller cloud. Because, again, each molecule is 2,000 to 3,000. So if you want to make 10 molecules, that's like a nice car. If you want to make 100 molecules, that's like a goddamn nice car, So 100 molecules gets you into like an affordable house territory, and, it gets worse.
Is there a way to make it in parallel to speed up the process?
Yes, but not with the money. The money is still going to be times that number.
Well, you can make, let's say, 1 million of them, right? But it'll cost $3 billion. There's not like, or let's say whatever, maybe there's some economy of scale, but his point is like, because there's just some like, it's like 9 months to make a baby, like there's some part of just synthesizing the molecule that's, takes time.
So would it mean that the big pharma companies are at a huge advantage comparing to startups because they're not really limited by money, and then they could...
They're still limited by money. Everyone is limited by the same. Money and creativity, or creativity and money. It's tricky. So big companies have their own chemistry. And we actually have our own, and it really helps. Because when you have chemistry in-house, it actually gets not quite as expensive as you think. It's still expensive, but also you get these nonlinear benefits. For example, it's faster because people who are working on it are they have skin in the game. They want it to go faster. They are really curious also. We're all scientists, so we also want to know. Again, this is my kind of classic harp on CRO. The CROs are good, and we use them, and we like them, and we pay them nicely, but they are not really you. They are working for everyone. They are not interested in any of it. They're just interested in doing a good job and getting paid. So we don't necessarily have the same interests as they do. End of story is that big pharma has advantages, of course. Its advantages is scale, the amount of funding available, the sophistication of research techniques available. The disadvantage is essentially the burden of bureaucracy and the creative power. So I don't think that people in big pharma are any less creative than people in small pharma, but the organization tends to dampen that. The risk-taking at large pharma is less easy than it is for small. Because what do we have to lose? Well, worst case scenario, we will not succeed. We'll all have to find another job. In a big pharma, the risk is a lot larger. So therefore, risk aversion, any big organization is by definition a little bit more risk averse than a small one. So,
but interesting that it's more and more in pharma that actually they mostly buy things that are already closer to, like they're not even, are they still really developing fundamental drugs?
Absolutely.
I just peripherally interact with pharma industry, just living in Boston and working in biotech. It seems like what's happened is that they've taken the really serious R&D and turned it into these almost like CROs for R&D, where you have these startups that are kind of made up of people that used to work at the big pharma. They do it in a startup where it's like you can take more risks, your career is more dynamic, and then if you succeed, the pharma repurchases all these people and all the assets and everything, and then takes it further into animal studies.
Mostly
or whatever.
The assets. Let's be honest, mostly the assets. The people are given money and
they can just go do it again, right? But I guess, okay, we missed your question. You had a question that we missed or something.
Oh, my question was essentially like, how long does it take on average to discover a new drug? Like in terms of years, in terms of the experiments needed?
To get to a drug candidate, if you're exceptionally good, about 3 years, more likely 5. A drug candidate means it hasn't been in people, but it has been in some animals. And then to finish the animal studies and go through the NDA process, another 5 to 8 years, if you're really good again, at which point you better be a large pharma, because for a smaller one, it would take longer. So we're talking a better part of 2 decades.
Wow. And then I guess after that you can get a patent?
Somewhere on the way.
Oh, somewhere on the way. So that guarantees like 20 years? Or?
So this is a game that most pharma companies learn to play, and most pharma companies play well, which is when to patent, because as soon as you patent, you're kind of on a timer. The more you can save of the patent life, the better. On the other hand, your competition is doing it too. So if they patent your favorite molecule that you spent 10 years developing before you, that stinks. Nobody gets bonuses for that. So it's very difficult. And you have to make clever decisions, and you have to operate on incomplete information. Because certainly pharma companies are not in the habit of sharing their progress with each other until it's done. Then they're very happy to share, of course, to illustrate their worth. But until it's done, it's secret.
So because the life cycle, the discovery cycle is so long, it's about 2 decades, you said, does it mean that right now there are a lot of good drug candidates and almost drugs which we don't know about, or some of us maybe do know about, which change our lives in next few years?
Absolutely. There's always a backlog. FDA has a fairly convenient but difficult to navigate website where you can look at clinical trials. Because once you enter clinical trials, generally speaking, a lot of the information is out there. You don't necessarily know what the molecule is, because clinical trials don't have to reveal. There's tricky rules there. Point being is that you may not find out what the molecule is, but you'll find out what is it for. And once clinical trials are completed, most pharma companies publish the outcome, and this is how you find out what happened. But there's a lot of stuff always in process. But again, it's fair to say that a lot of that stuff is also kind of a better working, improved things that have already been discovered. There's a big distinction between first in class and, for example, same in class or best in class, right? First in class, you're addressing something that hasn't been addressed before, whereas best in class is somebody addressed it, but you have a distinct advantage. For example, it's safer, or it's easier to make and cheaper, or it's more bioavailable and don't have to inject it, or any one of those things. You can inject it like once a month versus once a day, that kind of thing. But typically, it has to do with efficacy and safety. A lot of improvements are done on the efficacy and safety basis.
Can you imagine a world where a drug gets developed in 1 year? What would that be? How would that world look? Is that a possible world? What kind of technological advances would there have to be, or changes in the regulation? I don't know. What's that world? Let's say drug discovery on Mars,
Predictive power of human trials with minimal humans. That would be a very helpful thing. If you can develop a model that, based on, let's say, animal data, lets you decide what's going to happen in a person, and then with a minimum amount of human trials can make definitive and accurate prediction of what's going to happen in a larger population, that will speed things up. To do that, you of course have to prove that it works. So it's not going to be an instantaneous process. But I do believe that there are companies out there whose entire intent is to do that.
With software?
Software, Ouija boards, crystal balls, whatever. So it doesn't matter what it is. I'm just saying there's a requirement there. So that's one. Likewise, things that avoid animal studies and lengthy efficacy studies. See, some diseases, the progression of disease is slow. For example, fibrosis of various kinds. How do you emulate fibrosis in a test tube is also not entirely clear. How do you trigger fibrosis in a lab animal is not also clear. And several drugs in this arena have failed because they didn't transition. So they don't translate from cells to animals or from animals to people. And it has to do with the poor understanding of the underlying causes, among other things, but also the inability to trigger it in a suitable model. If you're developing a drug against, let's say, cognitive decline in a person, to test it properly, you do need multiple years just to see if people will stop cognitively declining or it'll get better. So there's just a degree of tenacity that you have to have. It's kind of like being a tree farmer, right? You can farm corn, that's 1 year. You can get 2 corn or maybe even 3 corn in some warmer climes. But if you're farming trees, you just got to get some patience.
And imagining that you have some unlimited resources, power, what would you do, like maybe to change the current status of the industry?
Well, if I had unlimited resources, I would give a lot more funds and a lot more cohesion to the current discoveries in the antibacterial, antifungal, antiviral arena, because I feel an existential threat coming on from that direction. Because we are very happy right now. Everybody gets pretty good. It's not too sick. That can change. So bacteria can be very, very sneaky. And let's not forget that a lot of the accomplishments of our era that we credit to healthcare have to be credited to the parts of healthcare that have to do with systems rather than drugs. The fact that we have clean drinking water, the fact that people generally avoid barfing into the same water supply as you're drinking from, the fact that we eat vetted foods, right? We have a Food and Drug Administration that actually goes out and checks that the meat is okay, that our clothes don't contain asbestos, that kind of stuff. That is not drugs, but it's equally important, if not more important. So paying more attention to that is a very good investment. And I would probably break down a lot more on the barrier side. So if pharma companies weren't afraid of sharing, for obvious and very reasonable reasons, if you can make it less difficult for pharma companies to share information between themselves and with academia, then those tiny moments of serendipity that is essentially at the core of almost every great discovery would be exposed a lot more. And so more people could notice that there is serendipitous things happening, as opposed to fighting against the current and just being lucky.
Are there any open source projects in the pharma?
I think there are some, but I don't know how real they are. I think there were several collaborations in large pharma. I just don't have enough information to tell you how successful they were or not. I would love to see more, but see, unlike code, it's just quite so much more expensive to get into this. So you can write a code on a broken laptop from 1990s, and you can still write reasonable code, and it'll run. You cannot do drug discovery on a broken laptop. It's just not doable. Now, you don't also need multi-million-dollar equipment. You can kind of compromise in the middle. So most startups don't get the bestest equipment. We get by on mid-range used stuff, and it still avails to good science. But certainly, it still costs hundreds of thousands of dollars total. So to give you an example,
just setting up the basic equipment to start doing work. Yeah.
So if you want to have a decent molecular biology biochemistry lab, so not chemistry, just molecular biology biochemistry, a little bit of biophysics, which is the foundation for sort of basic drug discovery, you need to spend, and this is coming from personal experience, you need to spend about $500,000. And that's just in stuff.
But you should mention that's literally the cost of one machine if you buy it new. So this is like if you're really good at hacking these machines, this is literally a factor of 10 less than the naive approach.
Is there something like, let's say in nanotechnology or semiconductor industry, we have some like clean room labs where you have shared equipment, so you don't have to buy that expensive stuff you can share with others?
I don't love the idea of sharing with others because, simply speaking, people do bad things. And then your stuff doesn't work when you need it to. So it's okay to have your own.
Yeah, the problem is that biology is even more black magic than cleanroom work. And so if something doesn't work and it's because the tool is shared, it's really tough. But cleanroom work has the same problem. If you're in a shared cleanroom, you end up, there's contaminated tools and all these things.
Yeah, there's definitely such kind of problems. But it seems like maybe on semiconductor industry, it could be like the processes are more controlled, so you can clean them out better.
There are companies that kind of claim to do this, like Ginkgo Bioworks. There are things like we're a foundry for biology. So there are like foundry type things, but they're not mature at all. So there's no like, you make a design, you send that, except for if you want chemistry, yes, but for biological stuff, not yet. But there's definitely, that's like a direction that's happening. There are also like these semi-automated biolabs that you can send them like, do all these experiments for me. And they'll run them all, but it's not really automated, like there's humans.
Well, you can send out any experiment pretty much to any CRO that knows its business, contract research organization, and they will do it for money. The difference part being they will do it for money, and then you will spend another unit of money to do it again. So I've always been a proponent of a hybrid model at a minimum, where you have enough of your own to ground the discovery in reality. But I've seen companies, I don't know how successful they were, I've seen certainly enough companies, and they're still out there, which are called pure virtual. So it's a bunch of very intelligent folks who, for various reasons, decided not to invest in physical infrastructure.
No equipment at all.
That's right. They just rent a space like this from whatever, WeWork or something, and they sit down and they design molecules on a computer. Then somebody in, say, China makes them, and then somebody in Japan tests them, and then somebody else analyzes the data, and they spit out that stuff. It still costs money. In the end, costs about the same, but different avenues of spending it. So now you're spending it teaching your competition how to be better.
Who is the best scientist?
I don't know.
In history. Who's your favorite scientist?
Oh, my favorite, probably it would be a tough split between Nikola Tesla and like Niels Bohr or one of those guys, the old physics types. But Nikola Tesla probably beats it.
Why?
Because he had application. He built things, and they worked. And also because he was quite ethical in his way. He dreamt of being good for people.
Interesting. Yet another, maybe one of our traditional questions. Do you have any recommendations for young scientists who are maybe just starting work in the field of drug discovery or who are planning to enter it?
My only one specific recommendation is to be as much of a polymath as possible, because drug discovery is a polymath business. If you're a pure chemist or pure biologist, it's perfectly fine. There will be a great career in front of you. But for purposes of personal enjoyment, if you understand what's happening across the disciplines, you'll just enjoy it more and possibly be more productive. There's always experts. Experts will not go away, and it's good to have and to be expert. But it's often the case that drugs can be, in fact, discovered by polymaths, people who know more than one discipline.
Is there anything that we didn't cover, like a story from your personal work or anything else that you think would be relevant that would be good to cover before we finish?
No, I think we went through an astonishing amount of stuff, and I actually apologize because my conversations tend to end up in various corners. Also, I would like to expressly mention that mistakes are all my own. I'm sure I've made them. I'm sure I've told you some stuff that probably upon deeper verification may not be fully accurate. But there's a lot of opinions that I've expressed, and those opinions are also mine. The moral of the story is it's a complicated world. We are complicated things that live in a complicated world. The biggest mistake that we as people can make is oversimplifying. Treating things and people as objects, especially people, is one of the worst mistakes that one can come up with, because we're not. We're much more than that. And just be careful making simplifications.
All right, Artem, thank you. This was very fun, very wide-ranging.
Fantastic.
I think we learned a lot.
Thank you. I've learned a lot from you guys.